Okay, so are we live, Isaac?
I don't see live right now. I'm starting. It's
Okay.
starting.
Isaac, feel free to keep talking so we know that we're actually live.
Yeah, I see it as live now, so you guys are good to go. Just yeah, go ahead.
Okay, so all right, thanks, Dr. Avi, for doing this. Some technical issues getting started, but we're streaming live now. So I'll just say a little bit about what we're doing, and then Dr. Avi, you can introduce yourself. I'll introduce myself, and then we'll start digging into the data. So, been wanting to get a live debate on the science because this is a very hot button topic, as everybody knows. It's very polarizing, and there's a lot of data out there. And unless you go through all of it, pretty in depth, you are not really going to get the full picture of what's going on. And it's easy to cite one article or thing that says this and another that says that, and we got to look at the totality of the data. So, Dr. Avi, tell us a little bit about you and how how you got into medicine and what your practice is like, and how you got into this kind of stuff, research and debating vaccines and that kind of thing.
Sure. So, hello everyone. I'm Avi. I went to medical school at Jefferson Medical College. I currently work in clinical research, and I have been getting into the debate sphere, as many people know. People in the Discord debate sphere will be familiar with this. And what brought me specifically to this was a mutual friend who requested that we do this debate. It's not my first vaccine debate. I've had other vaccine debates, but not in a live format. I've heard many anti-vax arguments, and I'm just not convinced by them. And I do seem to be convinced by the safety data and the efficacy data. So I want to, put these arguments to the test and to see if there's any. I know there are anti-vax that say the same thing about the pro-vaccine side. So I am, So I'm willing to meet whatever. I know there's like $100,000 thing or whatever going around. I'm willing to meet that too, and hopefully we can have this conversation in good faith, and hopefully we can base our views on the data and not on ideology. And yeah, I'll just leave it at that.
Okay, thank you, and I agree with most of you on that. I want to have a respectful debate because this is something that really gets people really riled up. And Isaac is on the line. We said you could moderate if things got bad, but hopefully we can just have a respectful discussion and look at the data. Before I introduce myself, I want to say that I don't fall under the label anti-vaccine. I don't think that that is a fair description of my views. I'm more just pro-drug safety, pro-informed consent. Have at one point in time was pro-vaccine, but had enough empirical evidence coming my way, stories of people telling me they'd been injured, that I had to look into it for myself. And what I found was not what I expected to find. And so I'm really excited to show this kind of data. So just a little bit of background on me. I'm a doctor of chiropractic, had a long career working as an engineer in the semiconductor industry before that, but came down with an autoimmune disease and, had to figure out a way to get well. And it ended up being a plant-based diet, mostly raw plant-based diet, that was able to heal me. And that just brought along a lot of revelations about how health actually works and how the medical system really isn't set up to treat autoimmunity and a lot of other chronic conditions well. And that's one thing that Dr. and Avi see eye to eye on. We're both vegans. So I felt like this makes it a little bit more of an amicable debate. So so I guess I will start off with my thesis is that, essentially I wanted to set the landscape here, because I think there's a lot of assumptions that people make about vaccines that aren't necessarily so. And so there's assumptions like, oh, well, the companies that make them, they don't make any money off of them. We have money to show that, Merck, who makes Gardasil, makes $2 billion-ish a year off the drug. The other thing is that the lack of liability. This is the main thing, and I can show you the 1986 law, which basically said that the United States passed a law that said if a vaccine product injures you, that the manufacturer can't be held liable for damages. That now, if like a lisinopril or a Viagra or a Zoloft or a Prozac injures you, a normal pharmaceutical that's not a biologic, that's not a vaccine, injures you, you have legal recourse against the manufacturer. But if a vaccine product harms you, basically you have to sue the U.S. government. And there's like a tax on each shot that goes into a pool of funds, and then the government decides, whether or not your injury is worth compensating for. And this fund has paid out something like $4 billion since 1986. But the fact is that, like, because the manufacturer doesn't have liability, they don't have a lot of incentive to make the product safe. So they can just make it kind of like appear safe enough to kind of get it out to market, and then let the damage be done and just kind of throw their hands up and say, hey, not my problem. So let's see, what I would like to do, Avi, if this is cool, is just start with the vaccine insert.
Yeah, so I guess I have a couple of things to say about what you said, actually.
Sure. Go ahead.
So the main thing to say here is that this is actually tangential to the debate proposition. So even if it is true, even if everything that you just said was true, it's compatible both with the Gardasil vaccine being safe. It is also compatible with the Gardasil vaccine being not safe. All of these things can be correct. You can have all those laws in place. And I haven't looked into those laws because they are tangential to the debate proposition, nor is it relevant in my view the amount of money that Merck makes from the Gardasil vaccine, because if it is safe and effective, I don't see a problem with them making money from the vaccine. So the safety of the Gardasil vaccine depends not on whether Merck makes money from the vaccine or how much money Merck makes from the vaccine, nor does it depend on the laws of various different countries on liability and lawsuits. The safety simply depends on the data itself. It doesn't depend on any of those other factors, and so those things I would simply dismiss as tangential position.
Okay, I understand, and the data is the data, and that's what we're going to go over today. But I think it sets the scene and just basically shows that there is an incentive to put drugs to market that are not safe when you don't have liability to do it. But okay, anyway, we'll.
But Ben, the issue there is, like, this is the same kind of argument that can be made about any pharmaceutical company. Any pharmaceutical company with even without the law is the separate issue, but even with the law, even having an incentive doesn't mean that. I mean, look, I can argue incentives. I can talk about, like, even with those things, they still would want them to be safe. They still would want to make sure that they're safe, because just from the bad PR alone, and just from all the negative. I mean, look, if they had the choice between, hey, do we want a bunch of, and I'll use the word anti-vaxxers, but do we want a bunch of them parading arguments around like this, or do we not? They would not want to do it. But again, this is just an induction. This is a loose induction similar to industry-funded studies. Industry-funded studying doesn't dismiss a study. It doesn't do it not by my account in nutrition, not by my account in pharmacy. It's about the methodology and the validity of what is being done in the study or studies. So I don't think this is relevant. I just think it's tangential to the debate proposition. It will be superseded by, you could set the scene any way you want, but it's superseded by the data. So we can just I think the right way to go from here would be to just get into the data.
Okay, I agree with you.
Actually,
All right.
before we even get into the data, we should actually just go over the debate proposition. So the debate proposition is
Okay.
Gardasil
Go
vaccine
over.
is safe. I'm sorry. Am I coming? Okay, sorry. Hold on one moment. Okay. Can you hear me?
Not too well.
Isaac, am I coming through for you, or is it just okay?
Sorry, I have to unmute in 2 places to talk. Yeah, you're coming through fine. I think it's just when you're talking over each other. So just try to give each other turns and it should be fine.
Okay. Okay. I'm switched over now. Can you hear me now?
Yeah,
Hello?
you're good to go.
Okay, cool. So I think the right way to go about this would be to look into the data rather than trying to set the scene with like, oh, this pharmaceutical company has incentive to do this or that. Arguments stand on their own 2 feet, not on the people making them. And so it's very important to look at the data. And really, we need to set the debate proposition, which was that Gardasil vaccine is safe. When I take that proposition to mean is that one thing I wanted to hash out, but
Hold, you cut out. So the proposition was that the drug is unsafe, or what is the proposition?
the Gardasil vaccine is safe. One thing I just wanted to clear up about this word safe, because one can draw that line of safety to an epistemic standard such that all pharmaceuticals are not safe. They can set a goalpost or they can set an epistemic standard, so essentially nothing is safe. And I hope that that's not something you'll do. I hope you'll have a fair goalpost here. I hope you'll have a fair epistemic standard for the word safe. So for example, if there's no symmetry breaker between Gardasil and any other injectable based on the safety data, then it wouldn't be, you wouldn't draw the line of safety such that everything is excluded. You can, but then we would have reductios. Anyway,
Okay.
do you have anything to say about that?
Sure. So what I would say is that what I want, my goal of the debate here is to present the data to the people watching to be able to make their own decision whether or not they can look at the risk and the benefit and decide whether or not that's an appropriate decision to make for them or their child. Because yes, I mean, obviously no drug is 100% safe. And so the question becomes like, is it safe enough that knowing like the totality of the data, I would do or not do, I would or would not get this injection for myself or my child. I think that's where we're trying to get to, because ultimately, in my opinion, like I'm pro informed consent. So I believe that people should know enough and then be able to make their decision. Like a doctor should present them with a diagnosis and a set of treatment options and say, here are the risks and benefits of each of these. I think you should do this one, but it's ultimately up to you. So that's right.
Yeah. No, I understand. I understand that view. I'm just saying the debate proposition is not whether laws should be in place for vaccines or not, or whether there should be informed consent. The debate proposition is the Gardasil vaccine is safe. So with respect to that proposition, whatever standard, all I'm saying is whatever standard you have for safety, that needs to be applied consistently, logically consistently, or else you're in either contradictory territory or you're in a territory where your line for safety would be so absurd that you would have to say everything is unsafe. That's my view on essentially virtually everything is unsafe. That's my view on where the data stands for the Gardasil vaccine at this point, and hopefully I can show that in this debate. So with that said, I think we can, just as long as that's clear, the point about logical consistency with respect to an epistemic standard. With that, as long as that's on the table and that's clear, we can proceed to the data.
Okay.
Now so before I interrupted you, sorry about that, by the way. Before I interrupted you, you said you wanted to start with the insert?
Yeah, I thought that was sort of the jumping off point for my argument to demonstrate that I wasn't satisfied with the level of safety that it demonstrated. And then, we have other data that I've sent you and you've sent me that we can kind of like hash out the details. But I feel like the safety study that's in the insert is kind of like the starting point.
Okay. I'm looking at the insert right now.
Okay. Let me pull it up so that because I want people to.
And Isaac, if you want to share it on, if you want to share the it on screen for those here, I'm linking it right now to you. I don't know if screen share is not.
Yeah, I'm not able to. No, screen share is not possible right now.
All right. Sorry.
But you can put it in the chat on the live stream, which will still be there after the stream, so people will be able to click the link as they're watching the video. So that's what I'd recommend.
Okay, so I'm in it on my live stream.
Okay.
Can you guys pull it up?
I have it up.
Okay.
Are you sharing screen on your live stream? Or are you? I'm just asking. The only reason I'm asking is the audience. It's just for the audience. If the audience can like follow along with us, or if we're just going to have the conversation.
Here's what we'll do. When someone wants to share data, just put it into the Discord conversation, and I will copy it into the stream conversation. So there's only one link there so far. Avi already put it there, but going forward, anything you guys share, just put it there. I'll copy paste it into the stream conversation.
Okay, is this?
And then at the end of the debate, we can, just compile a big set of links and put it in the description.
Just put it in? Okay.
Yeah.
All right. So I just want to make sure. So I'm streaming it on my Facebook Live so my people can see it. But I just want to make sure that we're all looking at the same thing and it's not like, well, here on page 4 it says this. Okay. So so just to reiterate for people watching, the package insert.
Sorry, Ben, could you throw it in the Discord conversation so that I can upload it for everyone to see here?
Oh, it's the same. It's the same link. He's talking about the package insert.
Okay. Yeah. Anything further, just throw it in the Discord. That'd be great.
Okay. Thanks. So I just drag the icon into the talk here.
Yeah, I mean, you can see the little typing area. You just type it there, and
you're
good to go. Yeah, just like any messenger.
I have locally saved on my computer, and I don't know where, or that I got them through emails when I requested their article. So is there a way to actually upload a file, or is it only?
Oh, yeah, that's just going to be a limitation. I think that what we're going to have to do is just compile a list after and put it in the description. But anything you can show on the go, you can put in the Discord, and I'll throw it in the description, or I'll throw it in the chat so people can watch. Yep.
So we got this one, and so just to explain for those unfamiliar, because I'm sure a lot of people watching are not healthcare professionals. Essentially, a package insert is basically the data that goes in when any sort of drug is in its box or in its package. It's like a fold out document that has all the information that the FDA requires is in there, and so including the testing that was submitted to the FDA for approval. So this is the data that was submitted to the FDA for approval. And so basically what we have is this safety study was done on, how big was the cohort? It was approximately 9,000-ish people. Okay.
Just to correct, just to as a point of, sorry to interrupt, Ben. Just as a point, this is the 9,000-ish people. It was 10,000. It was 10,000. Well, it depends if you're talking about women or men. It was 10,000 in the Gardasil. It's a compilation of various different protocols that go into the vaccine insert. So what they've done is there are protocols that are randomized controlled trials. They have combined them, or sort of in a sort of semi-meta analysis. And in the Gardasil group, there's about 10,000. In the for women. Between the ages of 9 and 26. For the placebo, there's about 9,000. So that's about that's 19. That's 19,000. And then for men, there's about 3,000 in the Gardasil group and 2,000 in the placebo.
Sure.
So we're 20.
So
it's about 25,000.
So I'm on page 4 to get started, and this is just showing adverse reactions. Within 1 to 5 days of post vaccination. So this is just looking at very like simple, straightforward, empirical adverse reactions? Was there pain at the injection site, swelling, erythema, that's redness, pruritus, I assume is pus, bruising? Okay, and it's broken down on page 40.
Pruritus, just Ben, I'm sorry, I'm really sorry to interrupt. Pruritus is not pus, so pruritus is itching.
Okay, fair enough. And yeah, feel free to intercede on technicalities like that if I make a mistake. I don't mind being corrected. And so we have the Gardasil group, the group who got the Gardasil drug. We have the AHS control, and we need to go into depth of what that is. And then we have the saline placebo group. So we have essentially 3 experimental groups, 2 of which are controls. And so the AHS, you can see, I'll highlight it here on my screen, but amorphous aluminum hydroxyphosphate. Okay, hydroxyphosphate sulfate. So this is an aluminum salt that is in the Gardasil vaccine, and then a subgroup of people got this injection as a control, as in they didn't get the virus and a bunch of the other things that are in the vaccine, and we can go over the full ingredients of what's in there, but they did get the aluminum. And then another smaller subset got the true placebo, which was saline, which is essentially salt water. So inert meaning like there doesn't believe there's anything that could interact with your body when it goes in. Is that a fair description of the 3 groups?
The one thing that is not mentioned here, and in good faith I will mention this, is that in the it's called the saline placebo, but it because it might as well be called a saline placebo. There are a couple of other ingredients in the saline placebo that is not salt water.
I noticed that, so please show that.
Wait, what?
Sorry, I said I noticed that. I was going to go into it later, but yeah, please expound.
Yeah, sure. So yeah, it's saline placebo. It might as well be called saline placebo, but technically speaking, there's 2 ingredients that are in there. One is 50 micrograms of polysorbate 80, and the other is I forget the dose of histidine, which is histidine, for anyone listening, is a natural amino acid that's found in the blood. It's found you get it in your diet, and it's a very, very low dose, far lower than what you would expect to ever consume. The tween 80, another name for polysorbate 80, is 50 micrograms. It's only been shown to be harmful in doses that are incredibly higher than that. To argue 50 micrograms would be harmful, you would have to actually argue that it would be more toxic than cyanide. That's why they list it as saline. There's another claim. Some people say it's boric. Some say there's borax in it, or boric acid. This is actually, there are some, I use the word anti-vax papers that say boric acid is also in the saline placebo. That's actually false. Boric acid was not in the saline placebo. The reason is because they simply didn't understand. They say that it is in the placebo, but it's not. The reason is because they simply did not understand boric acid gets in the vaccine in the first place. Boric acid is there as a byproduct of the aluminum adjuvant creating process. The design carrier, the design vehicle that is non-aluminum would not have boric acid. So if you might hear from some even in some literature that boric acid was in these placebos, that's not true. It was not in those placebos. The only 2 ingredients were other ingredients were polysorbate 80, 50 micrograms, and histidine, which is an amino acid. Okay. Sure.
And so just to clarify, sodium borate is in Gardasil, correct?
It's in Gardasil, but it was not in the saline placebo.
Okay. We're
it's 30, and it's 35 micrograms of sodium borate.
Okay. All right. So we're in agreement on that. Okay. And yeah, it is frustrating that, these things, given these names, and it's not what they say it is, which makes, doing the research on these things frustrating. I think you would agree.
So yeah, I mean, to be fair, in their publication, in their publication, they actually didn't. They actually did specify this. They specified that it was a carrier solution.
Yes.
When they
read that.
published this in a peer-reviewed journal. Yeah.
Okay. So we have these rather the more benign adverse reactions here. Okay. And so we see that, you know, for something like pain at the injection site, Gardasil had the most. The AHS control had slightly less, and then the saline placebo carrier solution had the least, but still had, you know, for example, in girls, 48%. So we go on to the next page, and then we look at, okay, this sort of just consolidates the data on page 4, if I'm not mistaken, just basically breaking things down into mild, moderate, or severe. And so then things get frustrating, shall we say, on page 6, because now we're going into slightly more severe adverse reactions. And so again, it's broken down between girls and women, but we've suddenly consolidated the two control groups. So now we're no longer showing Gardasil versus the saline placebo carrier solution. We're showing it versus a consolidation of the aluminum salt or the placebo. So they've essentially just like consolidated data in here, and that is suspicious to me. And so we see similar rates, right? Like we see a lot of these are, you know, about the same. The Gardasil slightly higher on pyrexia, fever, nausea, dizziness, diarrhea, vomiting, cough, toothache. That's always interesting to me. Upper respiratory tract infection, malaise, arthralgia, insomnia. All these things are slightly higher than the placebo, but part of the crux of my argument here is that an aluminum salt is not by any means a true placebo. And we're going to have to get into the science of what we do have on aluminum and its safety as an adjuvant to basically argue whether or not it's safe. But I just come up with the question of like, why did we not just test everything against the inert placebo? And why are we suddenly consolidating in the inert placebo data? Because it would be pretty interesting to see what it was broken out as it was on page 4.
Yeah. So you are. So and you can keep going actually with that. You can say on page 8, on page 9, you see autoimmune conditions, and they're consolidated as well.
Yeah.
Right. And so you saying now there is a way. The one thing I will disagree with you is that, well, there's a couple things I do. The first thing is that we need to go into aluminum and see if aluminum is safe because of this data. I actually don't agree with that. We actually don't even need to go into the technicalities of aluminum, because if all we need to do is split the groups up, we can just ungroup the grouping. We can take the aluminum control or saline placebo by splitting placebo being the non-aluminum placebo. We can actually just split them up and look at all the data. And if all the data, and if they're not statistically significant, and if they're not convincingly, like not even a great magnitude of difference, like less than 1%, then, and especially if they're not statistically significant, then I think that's game, set, match in a randomized controlled trial setting. We can actually just ungroup it.
Sure.
So what I did that's what I did. I actually went into the different protocols, and I actually just ungrouped them. I ungrouped the grouping. I took out I ungrouped the saline placebo, and I broke it down by the side effects, and I compared it to the Gardasil treatment group in the same study. Mm-hmm. And I also compared. What I also did is I ungrouped for the autoimmune conditions, especially. I ungrouped the aluminum control and carriers and non-aluminum control, and I checked to see if there was any difference. Statistically significant difference in autoimmune conditions between the aluminum and the non-aluminum placebo. and I tested it down the line, and there was not only was there no statistically significant difference, you would actually have to have, in almost every case, double the amount of the rates of, actually triple, not even double, triple the rates of autoimmune conditions in the aluminum group, and even one case in the non-aluminum group, and in just to reach statistical significance. Most cases it wouldn't even with that, and definitely not with doubling the rates. Also, not only was it not statistically significant, the difference was actually less than 1% in the total autoimmune conditions between the aluminum and non-aluminum controls. So we can go into the mechanistic data. We can go into the rabbits. We can go into the two papers arguing with each other about, aluminum and where should we set the threshold should it be based on oral and should it be based on it. I'm happy to go do all of that, but I think this actually supersedes it. The outcome is there's an evidence hierarchy, and randomized control trials supersede the mechanistic data. And so all we really need to do is, because here's the thing, Ben. So when we look at this data, this isn't the data that has been originally published. The Gardasil insert is just data that has been, again, like I said, compiled. They're compiling it from different protocols. They're compiling it from different studies. All of the studies are in that. I sent you a packet about, it was a very long 400 and some odd page.
Which I've read, and I agree we'll get into. Yeah.
Yeah. Have you read that in its, by the way, have you read that in its entirety or just protocol? Just curious.
No.
Okay. So what you can do is you can go into that in its entirety, and you can actually split up the AHS control and the saline placebo. And you can, so that's what I did. And I can share that. I'll share that in an Excel spreadsheet and I'll share my test for statistical significance. So I don't know if you can't really actually link. You can't link that into the chat, but I can share it for your purposes.
If you can Dropbox it and then put it in the chat box, that would be, that would be one way to do it.
Okay.
I have looked at Protocol 18, and I guess we can get to it eventually, but I just feel like that group that got the actual inert saline placebo isn't a big enough sample size to be able to extrapolate these things that are happening at a rate of 2.3%. So
okay, so it actually is the rates of 2.3%. They actually did pick that up. We know it is an adequate sample size because they actually saw it. So, for example, the thing that made and anyone can do a power analysis on this to show that this is true. But for the rates of things like, so the biggest thing that sticks out is arthralgia. And they did pick it up. There's no question about it. For 500 plus is more than sufficient to pick that up. And it would be more, anyone doing a power analysis on this, again, it's more than sufficient with the expected rates of arthralgia to expect you to detect a difference if a difference is actually there. In fact, you would have to, and the second point is, besides the point that I disagree with that statement as being true, the second point is, even if that was true, the rates of those things happening would be so low at that point. When you have 1,000, because it's not just 500 in the placebo group, it's 1,000 in the actual Gardasil group.
Yeah.
And yeah, it's the rates would have to be so low that at that point they would have to be significantly less than 1%, which they are in many of the grouped cases. You're talking about 0.1%, 0.2%, 0. And in order, even if it is a real effect, which it would be very weird if it was a real effect, because the baseline prevalence of all these conditions are actually higher. If you look at countries, and we don't have this data here because I didn't share it beforehand, but if you look at countries where you have all these autoimmune conditions, those baseline prevalence, even with countries that have lower vaccine schedules, those prevalences are actually higher than what we're seeing here. So it would be, so point one is that it's not true. Anyone could do, like, we can get a statistician to do a power analysis, and saying 500 is not enough. It's just a false. The other, it's in any accepted standard power analysis for any of these studies, like 1,500, randomized 2 to 1. No, that's fine. And you did see it pick it up. You saw arthralgias get picked up, which was the major, which was the highest frequency thing that could be adverse event that could be related to autoimmune conditions. They did pick up arthralgia. And not only was it not statistically significant, the difference would have to be so much greater for it to be statistically significant. It's not like it even got close. It wasn't even statistically significant at the p equals 0.1. Like, you could, even if you went to the 10%, not the p value being 0.5, 0.05, even if it was 0.1, it still wouldn't be statistically significant. And if we want, we can, even after the debate, get a statistician to do a power analysis on it. The second point is, even if that was true, it would still have to be, like, at that point, it would just have to be, the rates would have to be so low that anyone, like, at that point, like, gandering, like, oh, you need more people, like at that point, you'd be like, okay, well, the rate is going to be so low, even if you did pick up a meaningful difference at that point, it's not going to be, you'd have to question how clinically different it would be, the clinical relevance, because there's a difference between statistical significance and clinical significance.
Yeah, I
So
would agree that there's a difference in clinical and statistical significance. So when we have these, okay, so we're on page 8, we have these autoimmune diseases that people reported within, I think this trial was basically 6.5 months-ish, because they had injections at 0, 2, and 6 months and follow-up at 14 days, if I'm not mistaken.
For 14 days?
Yeah, after each injection.
Well, there was different, so there's different follow-ups. So for the expected acute adverse events, so there's different types of follow-up in these studies if you read the study design. So one type of follow-up you can do is a targeted follow-up for the expected acute adverse events of the vaccine injections. That you can do, that's fine, but that's not where it ended. They didn't only have a follow-up that's on the order of days. They also had follow-ups for new medical conditions that were developed that were long-term. They also had follow-ups for adverse events that were also long-term. They also had follow-ups for, and you can look at the schedule, actually the schedule of activities is not on the package insert. But in the data I sent to you, each of these protocols has something called a schedule. And the schedule of activities shows that they were actually doing it in protocol 18 specifically. The follow-up was for at least a year. And we can pull that up as well for the audience. But they assessed new medical conditions. They assessed any. So I mean, look, I've been working, there are things that you do as a standard practice in any clinical trial. You always, when you follow up, almost always check for new medications. You check for new adverse events. You check for new development of new medical conditions. All of those things are very standard. You can, on top of that, you can have a targeted, say, you got the injection. Let's have a targeted thing. Did you expect pain? Did you expect swelling? Did you expect pruritus, itching? Did you expect this, that? Did you that's a more targeted, because there are some acute adverse events that you would expect. Now, the confusion is people read that and think that that's the only thing they did. The confusion here is people read, oh, day 1 to day 15, oh my God, what kind of study is this? They only followed them for 15 days to see these adverse events. No, that's not what happened. They followed them in the randomized control study before they went to the long-term extension, which was 5 to 8 years, by the way. But even before that, while they were all still broken down into Gardasil and control group, they followed them for a year, at least. I think, and if I'm not mistaken, I think protocol 18 was actually longer than that. It was 18. I think it was 18 months.
I'm going to pull up protocol.
Are we on the same page about that? I'm sorry, I couldn't hear you, Ben. Can you tell?
Yeah, I mean, I'm pulling up protocol 18 here on my side.
Okay, so let me do that as well for me. Yeah, and I can show you where in the protocol that is. So let me actually get it for myself. it's a pretty big file. So, okay.
So I'm looking at the safety and persistent immunogenicity of a quadrivalent human HPV virus. This is the one with the 1782. Sorry.
Yeah, that's the safety safety and persistent immunogenicity. That's the one, right?
Yeah.
Okay. Yeah, that's the one. I'm just getting it pulling up on my screen. Sorry about the technical issues on my side. Yeah, I have it now. Okay, protocol 0018. And again, for those watching, we're going to try our best for the audience at home. We're going to try our best to have these sources. Maybe we can make a Dropbox or something, or a Google Drive or whatnot, and so that the audience can follow along when they watch this debate, and they can look at what we're looking at. Because unfortunately, I can't do a screen share right now with my my technical issues, but we'll have the sources available. So, protocol 18, yes. So let me just get on track to where you are. So what page are you on, Ben?
So just looking at even page 1 with the okay. So I'm trying to find.
So for me, yeah, it would be page 300. 300 is where protocol 18 starts.
Okay, I'm looking at the individual study, but I will pull up the 464 big file. Hold on.
Yeah, the protocol 18. Yeah, the individual study is important. That's published, but this protocol 18 on page 300, it has more information that is not just available in the published study. It has like a lot of supplementary information that is valuable for this discussion, such as the new medical conditions that were developed, and the follow-up that was long-term for the study. So I'm on page 300 right now.
Yeah, table 210. Wait, no.
I'm at
table 209.
Okay, table 210, correct. Yeah. So table 210, I see. Mm-hmm.
Okay.
Are you okay?
Yeah, I'm on 209 and 210. Yeah.
Okay. Table 210, I see. Is there anything you want to point out about is there anything you're looking for specifically in protocol? Oh, do you want me to show you? Oh, you want me to show you? I see. Sorry, I told you that. So look at table 211.
Okay.
Do you see table 211? Okay.
Yes.
So table 211, this is something called a schedule. Like, this is a very simplified version. They're usually I'm sure the real schedule of activities in the trial is more in depth than this, but this seems to be a simplified version of what's called a schedule of activities in a clinical trial. So on day 1, they were consented. And then on the consent day, they were given the vaccine. Remember, there's 3 doses to this vaccine. And they were given it a vaccine at day 1, at day 2, at month 2 and month 6. So that was the vaccine schedule for the Gardasil. So they had their treatment group have the vaccine, the actual vaccine, and then the non-aluminum placebo was also given at the same schedule of activities. And then they had a clinical follow-up for safety at each of those points. They also had it at month 4, month 12, and month 18. So after the last vaccination, which was month 6 vaccination, they still had a clinical follow-up 1 year later. Not only were they had 18 months of follow-up for clinical safety from the first injection, and they had 12 months of safety follow-up after the 3rd injection. What they also did, and this isn't mentioned here, they also had an additional 8 years of follow-up, at least 5 to 8, in some cases 10 years. But that was in a long-term extension where they weren't randomized to placebo anymore, that group. But they did keep in mind who was in the control and who was in the placebo group. So we can get into that as well. But there is at least in a fully randomized context here. Yeah, go ahead.
We also need to point out that people who received a year after these tests were done, and people who received the placebo were then told that they got the placebo and encouraged to get the vaccine.
Are you talking about at the point of long-term follow-up?
Yeah, my understanding is that after the 1 year after the last shot, the cards were put on the table and they told them, hey, you got the vaccine or you got the placebo. If you got the placebo, you're encouraged to get the vaccine.
Okay, so my understanding, and we can clear this up. My understanding was that this was blinded until month 18. I could be wrong about that. I'd like to know if
they were given the option to if they were placebo, given the option to get the shot, the actual drug.
At what point in time?
I have to look into the technicalities of this, but
yeah, because Ben, this is important because the important thing here is that vaccination was not part of the. I mean, the important thing is, like, I understand that they were encouraged to do that. I accept that for the long-term follow-up. If people were in the placebo group, I can only imagine they're encouraged to get the vaccine as part of their follow-up study, which was after month 18. Yeah. After month 18, they had what was this called a long-term extension where it's unblind. And so that I would understand. But if you want to make the claim, because that's a pretty serious claim that the participants were encouraged to get the vaccine. If you want to say it's after month 6, then I would need to see evidence for that.
No,
after month 18,
I'm saying month 18. Yeah, not
okay.
But I'm saying like the 8-year follow-up. I think that data is going to.
Yes.
I'm tenuous on that.
Of course. Yeah. Although I would say we can get into it, but I would say that the follow-up, the safety data on the follow-up, I do think was very, very, I mean, as far as any pharmaceutical goes, I think even if we did have a control with 0 serious adverse events, it would look pretty good in comparison to just about any other pharmaceutical. But right now we can stay with this one because we do have a year of follow-up, a year of long-term follow-up without any without encouragement to get the other vaccines.
Yeah.
And we have it, and it was blinded, and it was randomized, and it was and we have a year of safety follow-up, and we can look at so are we clear on that? Are we clear that we're not just talking about 15 days of follow-up? Because, like, I'll be honest with you, Ben, I've watched some of your videos on you don't have a lot, but you have a couple of videos on your concerns about vaccine. And one of the things I've heard you mention was that in these studies, they don't really do the long-term follow-up. They're only doing this follow-up for about 15 days or so. They're only checking for 15 days. And, I just wanted to clear that confusion up, Ben, because I see where the confusion's coming from. Don't get me wrong, because I see where you could read that and say, oh, there's only 50. It actually says right over here, day 1 to day 15. And you'd be like, well, what on earth is going on here? That's not a long-term follow-up. How can you do this? So I just want to clear that up because that's not actually what's happening. There is definite long-term follow-up, even in the randomized sense, at least what would be considered long-term by many different standards. So we do have at least a year of fully blinded follow-up there.
Okay. So let's get into the data here.
Yeah. Okay, sure. So I actually let me send my So if we look at so what you're looking for, I think what you're looking for, unless you have something specific that you wanted to point out to me, Ben.
So basically, when I looked at the one that was the 9-page version of this. Let's see, the one that safety and persistent immunogenicity. So that one, if I go and let's see.
Wait, are you on the PDF that I sent you, or are you on the publication?
I'm on one of not the original PDF you sent me, but I'm on the one that looks more like a journal study.
Okay, yeah, sure. I can pull that up as well. Okay, yep. I have it.
Okay. So yeah, a lot of efficacy data on here, which that's a whole other can of worms, because I'm not even going to get into it because we'll just create an argument. But presence of antibodies like may explain immunity, but like the average person dies of cervical cancer at age 58, so you'd have to have like a lot of follow up to show that like there's still immunity that much later on.
Well, look, Ben, we can have another debate. If you want to have a separate debate on the efficacy of Gardasil, that's fine.
Yeah, okay.
I am more than happy to have a debate on the efficacy and why I am fully stand behind the efficacy of Gardasil. I think it absolutely does prevent cervical cancer. I think the evidence is very clear. However, that's tangential to our debate proposition. We can have a separate debate on the efficacy. That's fine. And I'm happy to address any concern about the efficacy. I think that it's very, very clear that it's effective and it's durably effective, and it does prevent any based on the available data. It would be absurd to think it doesn't prevent cervical cancer, but that's a debate for a different time. And I'm only saying this, like I'm only grandstanding on this because you brought up the topic.
Sure.
But let's stick to this debate proposition. Yeah.
I agree, and I'm sorry that I brought that up because that was not the subject, and I appreciate you keeping me on subject. Because this could get in the weeds quick. So I guess I'm looking at page 207, which would be page 7 of the 9-page, the adverse experience summary, days 1 to 5. Yeah.
Yep. Yeah, and So what's the? Are you saying it's just because days 1 through 15? Is that what the issue is?
So in this, we show serious adverse events, 5 on the vaccine side and none on the placebo side. Am I correct?
Let me just take a look. Post this with serious AEs. One or more serious subjects with follow-up, percent number of subjects. You're looking at table 5, right?
Yeah.
I see with serious AE, I see 2 in post-dose 1.
Yeah.
And I see 3 in post-dose 2.
Mm-hmm.
And so I see 0.4% with serious AE and 0 with the non-aluminum placebo. Yeah, that's correct.
Okay. So, I mean, basically 5 out of so this is where my argument is that like the sample size
5 out of 1,000. Yeah, 5 out of 1,165 or 0.4%.
Yeah.
Also, the important thing, whenever there's an SAE, it's very important to not just say that there's an SAE. It's very important to go into what those SAEs were, because a lot of times the SAEs are so obviously unrelated to the vaccine that it would be absurd to even like have the conversation about it. So we have to go into what those serious adverse events are, and we can do that.
Okay, yeah.
I'm not based on what those SAEs. Yeah. Because based on what those SAEs were, I'm actually not. I'm not convinced that they are reasonably in any way, shape, or form tied into the vaccine.
Okay. I know we can go and I stuff like that. But
wait, I'm sorry, Ben. Can you just repeat that? I didn't hear.
I said I know they capture even things like people dying in car crashes and stuff like that. I get that. So let's find where to discuss what the actual serious adverse events are.
Yeah. So if they don't describe it here, that's fine. We can go into that long paper.
And if you find it for me, please let me know.
Yeah, they do describe it in the long paper. So, okay, 5 serious adverse experiences were reported through month 18, all of which occurred during the quadrivalent HPV vaccine series. None of those serious adverse experiences were judged by the investigator to be vaccine related. 2 of these serious adverse experiences occurred on 6 days and 2 days respectively after the first injection. Acute renal failure subject was discontinued from study, and insulin-dependent diabetes mellitus. 3 occurred 2 days, 11 days, and 3 days, respectively, after the second injection. Localized infection, anemia, and dysfunctional uterine bleeding and appendicitis. 2 subjects in the quadrivalent.
Okay, I think I found it.
Yeah.
It's the very end of the last paragraph on the page before the table.
Yeah. Yes.
You still there?
Yeah. Correct. None of the So
talking?
essentially,
I think he's
what happens when
I can't hear him.
Can you hear me? Can you hear me at all?
Can you hear me?
Sorry. I can hear you.
I can.
Hold on.
I can hear both of you perfectly clearly.
Okay. Did Avi just drop off and come back on?
He sometimes drops out and comes back, but I can hear him now. Can you hear him now?
Can you hear me?
Oh. Okay, that's not good. Avi, I guess maybe just leave and rejoin the call.
Okay, sure.
I'm gonna get some water. Hang on. Oh.
Oh no, it altered our positions. Maybe I need to leave and rejoin.
Can you hear me now? Is this okay?
Yeah, I can hear you.
Okay. So essentially, what happens is whenever there's an adverse event, what happens in clinical trials is there's an investigator who, in this case, did not work for the pharmaceutical company. That's important. So investigator is a medical doctor or a PI, what's called a PI, that oversees the patients. And it's the investigator's decision based on everything he knows about the patient, because the investigator has the most interaction with the patient, whether it's the medical history, whether it's the patient's likelihood of having things like anemia in the first place, whether it's the investigator, whether it's the subject's relation to the vaccine, so time frame, if they were fine, etc. All the symptoms, they are the ones who know the patients the most, and they give determinations of, depending on which protocol it is, depending on how it's written, they give various determinations of how likely an SAE is likely to be related to the investigational product. There are usually several options. They could be not related, possibly related, reasonable, possibly related. It depends on the various different protocols that are written. In all of these cases, the investigator, again, who does not work for the pharmaceutical company, determined that these were not related, that these adverse events. Now, they could be wrong. Sure, they could be wrong. It could be that we know something that they don't about the patient, but again, five serious adverse events in a sample size that is, twice as large as the placebo group. I'm not too surprised that there wasn't a serious adverse event in the other group when you have only 5. None of them were judged to be due to the vaccine. Some of them are very common. Some of them are very common in young people, young women especially, such as anemia, and dysfunctional uterine bleeding is very common. In fact, I'm very surprised dysfunctional uterine bleeding wasn't present in more of those people. I'm actually surprised to see how low it was. You had one appendicitis case. And you had
acute renal failure and diabetes.
Yeah, so those two we can hone in on. So one case out of 1,000 of acute renal failure, and the other case of diabetes mellitus.
I think all of them are on the table as far as I'm concerned, but we'll go into these two first.
Okay. Let's see if there's any follow-up on
And the acute renal failure, I believe she dropped out of the study as soon as it happened, right? That's what it says. Subject
discontinued with one with a treatment. Let me see. I don't know if were you seeing that the discontinuation because of the acute renal failure?
So in the paragraph above the table 5, where it says overall 5 serious adverse experiences reported in month 18, all of which occurred among the quadrivalent vaccine HPV.
Oh yes. Subject. So it's importantly to mention, like the subject recovered from the acute renal failure and discontinued from the study. So The adverse, so there was nothing, so the permanent ones I see are insulin-dependent diabetes mellitus. Yeah, so, okay. So the question is, so the obvious question here is were these were these serious adverse events related to the vaccine? Now, to be honest, we don't know. I would say even if we do know, which we don't. What we do for that is, again, we do what's called a power analysis to detect a certain level of adverse events. That's done in trial after trial. We see 5 out of over 1,000 in one case, and we see 0 out of 500 in another case. The question is, you actually can't do a statistical significance like a chi-square calculation on that, because you have 0. But even if we like triple, even if we make it like 15 to 1, and we just say there's 1 in there just for the purpose of having the calculation done, that's not going to be statistically significant. Even if you, and in fact, I think you even have to, yeah. And so this idea that, well, the sample size is too low, it's too low, we have to keep increasing, like, first of all, there's no guarantee that you'll ever find a statistically significant one. You could just end up getting in more for the control. But even if you did, it would be so low. The probability would be sub, again, it's not even 0.5%. It would just be so low that again, this is what I go back to, drawing this line of safety. When we talk about safety, if we set the bar of safety so high that we go from 0 point whatever percent, like, if we draw it so high such that we have that standard of safety, then you have to ask what's going to fall into the category of safety also by logical consistency, by extension. So if you say, 0, less than 0.5% happened in one group, not even statistically significant. Even if you tripled it and added 1 in, it wouldn't be statistically significant.
Okay.
If that's your bar for safety, you're going to end up saying a lot of things are not safe. You're going to end up saying a lot of things are not safe that people are going to look at you like you're crazy for. So again, this is all going to come down to logical consistency here. So looking at this data, my interpretation is you had 5 in out of 1,000, less than 0.5% out of SAE, not in the placebo group. Even if you multiplied it by numerous fold, even if you multiply it by 2 times, 3 times, I think even 4 times, and you did or even 5, if you did a chi-square test, you still wouldn't find a statistically significant difference. Or if you did any ever appropriate test for this study, you wouldn't find a statistically significant difference, even if you just had 1 in there just for the sake of having it and just started multiplying it several fold. Even if you increased, and even theoretically, if you did, it would be so low. The rate of having these things, which, by the way, in most of these cases, except for, I would imagine, except for the insulin-dependent diabetes, in most of these cases, especially, and it even mentions that in AKI, the subject recovered, that these weren't these debilitating long-term problems. So with the exception of perhaps the insulin-dependent diabetes, I don't think there were any other cases that actually did not recover from the SAE. So with that all said, I mean, it just seems very clear to me that this is like as safe as it gets. I mean, if you were to demand something safer than this, you're basically going to just end up saying that just about everything's not safe. You're going to be demanding that just about, like any farm. Look, if you have a standard to that degree, if you're going to have a standard where, like every pharmaceutical is just not safe, because you can keep making these arguments, Ben. You can keep saying, well, look, there wasn't enough. Maybe there was this point whatever percent of a adverse event that was experienced. Maybe there's like point whatever. If you can keep raising the bar and raising the bar further and further, and say, well, it wasn't different, but every time we see it in something not different, well, let's get more people. Maybe we could find something. Like, it looks like we're grasping for straws here at this point.
Okay, and I think I'm glad we got to here, because I think this is like basically where we disagree, and okay. And I understand all of your points that like the drugs can only be so safe, that any intervention like has its limits of safety. Like someone could like walk into a doctor's office and slip and fall onto a wet surface and crack their head open and die, like right there. That's like could happen, right? But I see where you're coming from, and I respectfully, very strongly disagree. I think that, okay, the fact that we have to add severe adverse events into the placebo group to even get a statistically significant effect should tell you something. That that all the severe adverse events are happening in the drug group, and we're basically 5 out of 1,000. So we're saying, well, it's really not enough data to make a statistically significant thing. To me, the answer is that you definitely need more data, especially when in the other groups, we go back to the insert in the other protocols, you tally up everything else, the rate of systemic autoimmune disease is 2.3%. So this
hold on, Ben. That's a different conversation. We could get into 2.3%. We can, but let's just stick to the SAEs. That's different than SAEs. So, is there anything
explain how
say after
this is the autoimmune disease is listed on the package insert is different than a SAE? Because
SAEs have different criteria. SAEs have criteria such as either, generally speaking, they either cause something debilitating either through hospitalization. So if someone is hospitalized for a period of 24 hours or more, that is an SAE. If it's causing some sort of debilitating condition that's long-term, it's an SAE. There's different criteria per protocol on SAEs, but they are not just having an autoimmune condition doesn't make it have an SAE. That's why I looked into specifically those who developed the autoimmune conditions and not just those who developed SAEs. I do have more details on those SAEs.
Okay, and SAE is it stand for severe or systemic?
Severe adverse event.
Okay.
Severe adverse event.
Okay.
Yeah.
So I think this is good that we're having this discussion. I want to finish up my thoughts on this, and you're saying that okay, even if we got more data, it's real, there's only a certain limit of safety. But I feel like people need to know this, that there's essentially a 1 in 200 chance of having something like acute renal failure or diabetes type 1, that we don't even understand the mechanism of how diabetes type 1 happens, right? We can't predict who's going to get it for sure and when, correct?
We couldn't even say that, Ben. We couldn't say that there's a 1 in whatever chance of having that, because again, these things were so low that you wouldn't be able. Again, there's certain confines in we need to show certain statistical significance. And again, what we do this is not we don't approach statistical significance. We don't approach whether we have enough of a sample size by looking at, well, we found 5 out of 1,000 here in SAEs, and then we had 0 in the other group. That's not how it's done to determine whether we have enough of a sample size. No statistician would agree with it. What's done is a power calculation that's done prior to the trial. It's done a priori, and what it can do is it can detect. It is a power analysis that can detect. Okay, is our sample size large enough for what we would expect? Those are typically done prior to the trial. They're not done post hoc. Post hoc. I mean, any statistician will tell you post hoc power calculation are very, very, very problematic. This is not how we determine that we didn't have the appropriate sample size.
If you're saying that we can only do a calculation prior to we do the experiment based on what we expect, aren't we doing a scientific experiment to see if
it almost it's they're done at I'm almost certain that they're done a priori and in fact you probably wouldn't be doing it scientifically if you did it post hoc. Post hoc, and I can have any statistician correct me on that. I have some stat friends who may be watching this, but everything I've read from the statisticians that I've been following, they time and time again they've been railing against post hoc power analyses. But I could be wrong on that. If I am wrong on that, I'll make a correction.
Okay.
I have a.
Basically, I feel like we're doing the experiment under the assumption that anything we don't like, we just say is then thereby statistically insignificant because it wasn't what we expected.
Wait, what? No, that's not what we're doing at all. It's just that the things have to be set in advance. So the power calculation is set before we get the data. It's very important. The things, it's like inclusion. I think it's like inclusion exclusion criteria. Inclusion exclusion criteria, you can't just do a meta-analysis and just have inclusion exclusion criteria that you're going to do post hoc, because then you're just setting it. Then you're doing something non-scientific. You're setting something because you want to say something. Yeah, it's the same thing here. If you were, so for example, if you were to be someone who wants to say vaccines are not safe, if you do the data first, if you get the data first, and then you set your threshold for power second, what you can do is just this. You can get data, and so long as there's at least one in one group and not the other, and not zero in another group, you can always keep saying, well, there wasn't enough people, there wasn't enough power. So it's the same unscientific post hoc rationalization for a position. That's why power analyses should be done a priori, prior to the study. But anyway, I'm going into
an entirely new study, because this is a drug that we're basically saying should be administered to every person at this age, and we're testing it on these 1,700 people, and we get a 5 in 1,000 effect, and we basically say, oh well, based on our math, it's not.
Well, let's actually go into the details of those 5 actually. And I would, even if it was 5 that could be clearly related to the vaccine, which I disagree with, I would actually think that would be fine. But if you actually look at the acute renal failure, so it's important to go into the details. If you go into the details, it actually seems pretty clear that they're not related to the vaccine. So where I am, and you can follow me on the long page here. So I'm on page 327.
Okay, let me get there.
So for subject, yeah.
Okay.
So you would think, if you just looked at the publication, Ben, you would think that these are related to the vaccine, because he got the vaccine here, and this and the renal failure happened. Oh my gosh. But if you actually look into the story, you see that there was a lot more going on there than getting the vaccine, and there's things going on that actually are more known to cause renal failure. So subject number AN71340 he actually broke his finger 5 days before his first dose of Gardasil. He required surgery, and then he developed renal failure. At the time of the operation, he received sufentanil citrate, lidocaine, tetanus toxoid, bupivacaine hydrochloride, lidocaine ketorolac, which is the ketorolac is really what we're looking at here, because that's known. An anti-inflammatory, renal impairment seen after long-term use, and dipyrone. So ketorolac specifically, that is known to cause acute kidney failure, AKIs. So it's not just that he only received the vaccine and then he got renal failure. That's not what happened. He broke his finger before the vaccine. He required surgery. He got medications known to cause renal failure. Then he got renal failure. That's why so this is why it's important to have an investigator looking to see the details of what's going on. Like, oh, was this related to vaccine? No. He specifically received intravenous. I'm pretty sure it was intravenous, like a lidocaine ketorolac. Which is, again, he received medications known to cause renal failure, then he got renal failure. And then if we keep going to the next adverse event, the next subject received the vaccine and experienced abdominal pain 3 days after receiving dose 2. She was treated with acetaminophen and ibuprofen. The subject was diagnosed with appendicitis 4 days after receiving dose 2. She was admitted to the hospital for removal of the appendix 7 days after receiving dose 2. The subject recovered and was discharged from the hospital 8 days after receiving dose 2. The reporting investigator determined appendicitis was probably not related to subject vaccine placebo. So we can put that as a maybe. So the first one, I would say definitely not. First one, we can clearly say this is attributable to a known medication that causes renal failure. The subject developed the renal failure. He developed it after he received medication specifically well known to cause renal failure. So let's take one. I would take one out of that 5 right away.
Okay,
I would say 2 is. Yeah,
okay. Go ahead. Keep going.
Yeah. So now we're down to 4. Number 2 is a maybe. The third one, the subject with insulin-dependent diabetes mellitus appeared to have an elevated glucose on the day of vaccination. So before even before they actually got the vaccine. And hemoglobin A1C was reported as elevated at the time of vaccination. So this is important. Although, quote, it may have been the upper limit of normal. So this is really important because the hemoglobin A1C is a measurement of how your glucose has been doing, prior to for about 30 days prior. So the hemoglobin gets glycated with glucose, and it's a measurement of how your glucose status has been over the last month or so.
Mm-hmm.
And it's important to note that this diabetes. Is that my headphones killing out? Can you talk for a second, Ben?
Yeah, I can hear you.
Okay, good. Okay, so this is important because we have data showing that this subject was having troubling blood sugars before they even received the first dose of the vaccine. So that's obvious.
Well, they appear to have an elevated glucose is not scientific.
No. Hemoglobin A1C.
Okay.
Hemoglobin A1C.
So are they drawing their? I just don't understand how they get this data point.
So in screening, very often in screening, they draw safety labs. That's again, like what I told you, remember with the schedule of activities? With the schedule of activities, in almost every schedule of activities I've seen, it's much more detailed than this. This looks like an abridged version of what we saw in the schedule of activities. So it's very common. What I'll tell you as someone who works in clinical trials for 3 years now, you almost always have a screening visit where you draw safety labs. You draw labs just at baseline, either at screening or at baseline, to compare the labs, what the labs look like afterward.
Okay.
The key thing that's standing out for me here is the hemoglobin A1C was elevated. It may be at the upper limit of normal. I understand that because different criteria would either be high or upper limit of normal depending on where your cutoff is. I get that, depending on which laboratory you use. But the thing standing out to me here again is if hemoglobin A1C is a measurement of what your glucose is 30 days before, and at least on some criteria, it's high, then that indicates to me that this person was having trouble with their glucose even before they even received the first injection, when we're talking about insulin-dependent diabetes. So that already is like huge. That's huge for me. That already is like, okay, I would remove that one. I would go down. We're still at 1 out of, I've taken off 2. We started at 5. Number 1, received a medication known to cause renal failure, then got renal failure. Mm-hmm. That's off. Number 2, the glucose was high. The hemoglobin A1C was high before they even got the first dose. So You're telling me the diabetes was because of the first dose? That's off for me. So let's keep going. The next subject had dysfunctional uterine bleeding and anemia, which may occur near the onset of menses. Okay, that's a maybe for me. So right now I'll put 2 as a maybe. 2 of those 5 are definitely not. And then let's go to the 5th one. This subject developed an infected toe and pain in his right toe and was taken to the hospital 2 days after receiving dose 2. Laboratory tests performed revealed an elevated white blood cell count, neutrophil increase in C-reactive protein of 127. The subject recovered from the pain and was discharged from the hospital with instructions to soak the toe in soap and water 3 times a day. Repeat laboratory tests performed 5 days after receiving dose 2. The subject recovered. The reporting investigator. What? I'm sorry, Ben. We were going to say something?
Yeah, finish and then I'll ask my question.
Yeah. The reporting investigator determined that the pain in the right toe and infected toe were definitely not related to the vaccine placebo. Okay, what's your question?
So, CRP of 127. Mm-hmm. I'm not pretty sure on the units. What normal is, like, the threshold is 1.0. Is that the same units or is that?
Yeah, I can check the if you want the actual limits.
Because if I know what I know,
those units, yeah,
like doesn't make sense.
Yeah, so the range of normal CRP, yeah, upper limit would be 1. Yeah, I mean CRP. Look, CRP is. Wait, let me see if that's in milligrams per. Let me just make sure about the units about that. So they're reporting the units in milligrams. Oh, that's why, because they're reporting the units in milligrams per liter. It's not. They're not reporting the units in milligrams per deciliter.
Okay. So
yeah. So I can convert it for you if you like. Yeah, I can convert the unit.
Thank you.
Milligrams per liter. Okay, 127 milligrams per liter per deciliter to milligrams per liter. That comes out to liter. $127 milligrams. Oh, we want to convert the other way. Sorry, I'm so sorry. We want to convert milligrams per liter to milligrams per deciliter. So that's fine. Let's do that. And then we get a CRP of 12.7. Does that make more sense then? Yeah.
Yeah. On the same page? Okay. Look, I knowing what I know from looking at labs, and I'm mostly looking at chronic conditions. 12.7 is absurdly high, especially for a child.
Yeah, but it's not. But keep in mind, Ben, that's actually not absurdly high in acutely infected individuals. You don't. It's not uncommon to see that in patients with acute infections. So CRP is it has a high sensitivity, if I'm not mistaken, but doesn't have a high specificity. You can see very high CRPs in acute infections. That's not uncommon for that. That's not unexpected. Now, I agree. If it's just a autoimmune or autoinflammatory condition, if you're dealing with that, that's pretty significant. But if you're dealing with an acutely infected patient, I wouldn't be surprised at all if you see a CRP of 12.7. So it seems to me like this. Okay, so someone developed an infected toe. I wish we had a little more history to see if we stubbed his toe or not. I don't see how the vaccine, would just randomly get a toe infected, and he recovered from it either way. He developed the toe infection, he recovered from the toe infection. Okay. So here's what my take on is of these five. Of these five, two are clearly, I don't think any reasonable person would say that they're related to the vaccine. One of them received a medication specifically known to cause renal failure and then developed the renal failure. To me, that's off. The other subject had evidence of diabetes had evidence of elevated glucose before they even received the first injection. So to say that the diabetes was because of the injection, no, that seems off. So we're down to three. And then the toe infection seems like the only two I think that are maybes for me. And this is only maybes for me just because I can't explain otherwise. Or not just I can't explain otherwise because they don't have a clear explanation for why it happened, other than just to say that these are common things that happen. The dysfunctional uterine bleeding would be one, and then the appendicitis, which again, there's no, even those I would argue, but even if we go down to 2 or 3, if we redo the calculation, do you know what the percentage is going to be then?
Yeah,
if we disagree on those.
3 out of 1,000, 2 out of 1,000, 1 out of 1,000.
Well, more than 1,000. It's going to be 1,000 and 1,179, I believe. Yeah, 3 out of 1,179. It's going down. Yeah, it's going to be 0.2%. Now, if it was 2, if it was 3, if it was 2, which I think it would be 2, it would be 0. Be 0.16%. 0.16. So look, we can keep getting the percentages further and further down, Ben. But here's the point I keep saying. At what point? What's your goalpost? I just want to know what the goalpost. What is your goalpost at which you would say, okay, you know what? We actually have enough here. We have 0. Even if we had a magic control group that was perfect and it had enough and it had 0. At this point, you see 0.0169% had an SAE. I don't know what more to give you, Ben.
But we have a bunch of other data. If we, all these other studies that are not done against an saline placebo that show pretty much parity in the Gardasil and the AHS, and in there we're at a 2.3% rate for autoimmunity. So wait. That's a slip, Ben. That's a slip. Now you're going from SAEs to autoimmune conditions. We can talk about that too, Ben. Okay. We got to stay focused here. Yeah. So now we're on SAEs. If you want to move on to autoimmunity, we can do that too.
So with respect to SAEs, because the other data, if you're going to bring other, you can bring other data from SAEs if you want to show parity. That's fine. But and we can talk about that, but if you want to go to autoimmune conditions, those are not necessarily SAEs. Those are different things. With respect to the SAEs, the SAE rate, when you actually look at what happens with those SAEs, I would put it at tops around 0.016, maybe 0.02%. These are levels that I don't think almost anyone would be concerned with when we're talking about pharmaceuticals. Almost every pharmaceutical is going to have. If you did a study on just about any pharmaceutical drug, Ben, you are going to see rates like that. Almost every time, you'll see this 0.0 whatever percent chance of having an adverse event. So with respect to SAEs, I'm not convinced, and I don't think anyone reasonable would be convinced. Now, if you want to go on to autoimmunity and make the case for that, that's fine. But right now, yeah. What? Sorry, you want to say?
I was agreeing with that we should go on to autoimmunity. I think that I still hold my point that on the SAEs we need more data. That 2 out of 1,000 is just like it's enough to show that we need to look into this more. You disagree. I think we agree to disagree on that because we've been hammering away at it.
Well, the only thing I'll say to that, Ben, is if that's the point that you're making here, if that's the point that you want to make, then you need to be consistent and apply that to just about every other pharmaceutical as well. So if you're saying we need to look into it more before we can say it's safe, that would probably apply to just about everything. It may even apply if you did a study on apples, you probably would see that as well. But even forgetting apples, because I don't want to get into the whole appeal to nature fallacy, because which is true. No, Ben, it's absolutely true. People who eat apples, you'll find 0. whatever percent of them end up having an SAE. Like, okay, fine. It doesn't mean the apples are causing the SAE. But again, like just about every pharmaceutical, especially if you looked at this, you'll find again, if you have thousands of people, 1 or 2 are going to have an SAE. Sure, like this just happens. It happens because SAEs happen. If you want to say that at that point you need to look into more, you can have that position. You can have that, but you have to have that as a consistent position. Which means
am I inconsistent with it somewhere else?
Yeah, it's consistent with all the other pharmaceutical products that also have SAEs at a rate of 0.1% or greater. Any other pharmaceutical that I can show you that has SAEs of 0.1% or greater, then you would be like, oh wow, we need to really look into this more. So you can have that standard of safety if you want. But the reductio on your view, that comes with a reductio. The reductio on your view is you've drawn the bar of safety so high that you're in a position where you're going to say everything is not safe until we look into it further. Just about every pharmaceutical, you can look into this, just about every pharmaceutical you look into, you're going to be able to make that same statement if that's your bar. So this is why, again, I'm going to try to, I'm actually at this point, I'm going to actually just ask you for a goalpost. What is your bar for safety? What do we need? Give me the numbers, Ben. What would you say is needed? What percentage, what number does a given metric need to be in order for you to say, okay, you know what? Most likely, I'm pretty convinced at this point that this is as safe as thing, considering, how everything has side effects, but this is pretty safe. What do you need to see? And then we'll go into the logical extension of that.
Sure, fair enough. Fair question. So my thesis is essentially that there should be informed consent where people know this before they're given the drug or mandated to have the drug and make their own decision. You understand that based on my license and everything like that.
I got to stop you because you're not that's not an answer to the question. So the question wasn't about legal systems, it wasn't about informed consent, it wasn't about any of those things. So the question was specifically, so remember, the debate proposition is not whether there should be informed consent. The debate proposition was not whether there should be in people being informed or whether there should be laws around this or not. The debate proposition is the Gardasil vaccine is safe. Now, my question to you, because I provided data showing that the SAEs were so low when you actually look into them that it would just match just about anything that's being safe. Now, my question, you seem unconvinced by that. So my question to you specifically is what numbers do you need to see for what given metric for you to say, you know what, it's safe. I don't want to hear anything about, people need to have informed consent. That's a different topic. I agree, whatever. I don't want to hear about, people need to know the data, there shouldn't be laws. Okay, none of that is relevant to the question. What I want to hear specifically is a metric or a set of metrics and what those numbers need to be for you to admit, okay, you know what? I've seen enough. It's very, very likely that this is safe. That's the question being posed to you.
Sure. And I think that is not easy to give a black and white answer. So what I would say is that if the proposed benefit is greater than the risk. Now we have the chance of dying of cervical cancer, as I understand it, is something like it's the 20th most common cancer. And so if the risk of a serious adverse event is considerably less than the risk of the purported benefit, then I would see. Here's the thing: I really can't, because I will not say whether someone should or should not receive a medical intervention. I can't.
That's not what I'm asking, Ben. But Ben, that's not what I'm asking. I'm not asking whether you're going to say someone should receive a medical intervention. Nor was that the debate proposition. The debate proposition was just, on your view, is Gardasil safe? And my question to you is, what numbers do you need to see? Give me the metrics and give me the numbers for you to be able to say, on your view specifically, not your view about what other people should do, not your view on whether the law should be, your view specifically on the proposition that Gardasil is safe. What numbers do you need to see to say, you know what, I'm convinced this is pretty safe? What's your goalpost?
I don't feel like I have sufficient data to be able to make that decision here, which is the crux of my argument. So at what point would I say it's safe depends on it's too complicated, because what are these adverse events? Are they going to be long-term and permanent? Are they going to be temporary?
You can specify
too much to quantify
them. You can specify them, Ben. You can specify all of that. Again, you don't need data to have a goalpost. You have these goalposts. You can say, like, look, here's all the metrics. Here's how long. Here's the ones that last this long. Here's the ones that last this long. Here's the ones that have this degree of debility. Here's the ones that result in death. Here's what I need to see. Here's what the numbers need to be for all of these. You can bake those all into the cake. And I just want to know what your goalpost is. Because, look, the reason I'm pinning you on this, Ben, is because at this point in the conversation, I wonder if there is any data that can change it. I wonder if any data exists or could exist at this point, or would you just keep moving the goalposts? So if we have, for example, I'll give you just a case, if we have 1,000 more on each side, and it turns out that it's 5 SAEs on one side and 4 SAEs on the other side or something, are you going to say, well, there was one more in this one, and then be like, we need more, we need to look into it more? Are you just going to keep moving the goalposts? So I need to know what your goalpost is at this point.
Sure. And if there was, if in theory, like you said, happened, where we, increased the sample size, and now 5 people in the vaccination group had SAEs and 4 people in the placebo group, that would change my views significantly. But we still have 0 in the placebo.
Yeah. What my question is, well, again, you can have, because the other possibility here is, Dave, that you can have an effect that is. But sorry, I keep calling you Dave. I don't know why. I just had a conversation with somebody. Ben. Ben, can you hear me, by the way? Because you're cutting. Yeah, Ben.
Yeah, I can hear you. Yeah.
Yeah. So the other thing that could be is, it could be that the vaccine does carry some very, very small risk. Ben, it could be that there is some really, really tiny risk. But again, the point is that even if that was the case, even if that risk was there, that in order to say that's not safe, there are so many other things you're also going to have to say it's not safe because of that risk. So my question to you is just, let's say there is some risk. Let's say there is some risk that's greater than the placebo. Because every drug probably has some risk that's greater than the placebo.
Sure.
When we get 0.016%, when we get something like that, At that point, I'm going to ask you, how low does that number need to be for an SAE? How long does that, and we can do it for everything. We can do it for autoimmune conditions, we can do it for SAEs. Right now we're on SAEs. What's your goalpost for SAEs? How low of a percent does that need to be for you to say, okay, you know what? Yes, there's some like very small increased risk compared to the placebo, but like that's for every drug. This is low enough so we can say it's probably safe. What's your goalpost?
I don't have an answer for you. I'm sorry to disappoint, but
so well that's important then, because here's the problem then. Like the problem here is if you don't have a goalpost, then what you're saying is that you actually don't know what number would convince you. So let's just say, okay, we did the data and it was 0.00000000001% above the placebo. Would you say that's a difference that, you know what, I think it's pretty much safe enough. I think that's fairly safe. Or would you say, you know what, I don't know, I can't give you the goalpost. We may need more data.
I feel like you're taking me down an archaeological rat hole.
Wait, what?
I said I feel like you're taking me down a rat hole.
No. This is no. This is important. The reason I'm taking you down this pathway is because there's a reductio on your view here. Your view entails something that is a reductio ad absurdum. Your view is that I don't know of a goalpost. If you don't know of a goalpost, then that also entails that if you don't know at all, that also entails that you can have a placebo that's 0.0000000, et cetera, 1% higher, and you would say, I don't know if it's safe. If you have that view, that is something everyone is going to find deeply unconvincing. If you don't have that view, then it's not true that you don't know. You do have some idea of what your goalpost is. So then you would just be in a logical contradiction. So it's important. I'm not trying to take you down a needless rabbit hole. It's important that you realize when you think, you need to think logically consistently about when you evaluate what it's all epistemic consistent. Your standard for safety, whatever it is, needs to be consistent. So my question to you, if you're telling me that you don't know what your goalpost is going to be, I'm just going to ask you, let's say it's 0.0000001%. Let's say it's that low. Are you going to tell me, you know what, I still don't know, we need more data?
No. And my problem
okay,
is...
so then you don't know, Ben. Ben, then it's not that you don't know. It's not that, then when you tell me you don't know, it's not that you have no idea. You may not have a range where you don't know, but you don't have it's not that you can't give any answer at all. That's a contradiction. It's a contradiction because you did give me some idea. You can't say you have no idea of where the goalpost is because you just told me about some idea of where you had the goalpost. So now it's just a question of where that range is.
Okay. I mean, but my retort to you is that if you can't quantify just lump in all adverse events as equal, like you can't quantify this risk.
I agree, which is why I'm giving you the option to stratify it out as well. You can bake that into the cake just as well, and I can ask the same question. So that's why, by the way, I specifically went into those adverse events, because right now we started off at 5. And when we looked into what actually happened, it was clear that a good chunk of those 2, if not 3, were clearly not related to the vaccine. People would have to be crazy to believe it was. There's like hard evidence that it wasn't. So that's why I agree. Not all AEs are the same, and not all AEs have the same for other reasons. But we can bake that into the question, Ben. We can just say, Ben, for what different types of AEs does a given percentage difference between the drug and the placebo need to be for you to say, you know what, I'm pretty confident that this is safe for as far as a drug can be. We can bake that in for autoimmune conditions. We could bake that in for debilitating conditions. We could bake that into the conditions where people experience it for a few weeks and recover. We can go through all of these. I'm not requiring you to say that all SAEs are equal and give me a number for all SAEs. I can ask the question granulated. But the point is, I need a goalpost from you, because if I don't, then we're not having a conversation. My interlocutor here, we're not having a conversation by which I'm convinced that there's anything that would change your mind. Because look, I'll just give it to you this way. There are things that I can give you my goalpost. there are things that would change my mind. If there is statistically significant difference between the vaccine and the placebo, if we were to find a statistically significant difference and it was clinically significant to the degree, say, I can give you, say, 5%, I could just throw that number out there right now, a 5% difference, or even a 2 or 3% difference between debilitating SAEs, whether that is permanent renal failure, or whether that is, death, or whether that is, or whether that's paralysis, or what I can go on. See, I can give you these things, and I can say, okay, look, if we actually showed these things, I will come onto your side, Ben. I will step over to your side, and I will be like, hey, we need to really work on these vaccines. I have a goalpost that I can give. It doesn't seem to me like you can give me a goalpost. And that's huge here, because that tells me that there may be nothing that can change your mind. You may not be operating scientifically. So that's why I'm asking you for a goalpost.
Sure, and I can't provide it. Obviously, I've said that. So you said that there's no statistically significant, correlation between, these serious adverse events. But you said that in order for there need to be to do a chi-square analysis, you would have to have at least one.
That was for the autoimmune, Ben. That was for the autoimmune. Just real quick, just to correct you, I said that about the autoimmune conditions. And what you can do is you could group the autoimmune conditions together for both, because it was enough to pick up autoimmune conditions. It didn't pick up no autoimmune. There were autoimmune conditions that were picked up. Mm-hmm. So what I did was I did a chi-square analysis on all the autoimmune conditions that were picked up, grouped to the total autoimmune conditions compared to the other one. But the key, and that you can do a chi-square analysis, and it was not only not statistically significant, it wasn't even close. Even if you set the p-value to 0.1, it still wouldn't be statistically significant. Okay, so it's just a complete wash. But here's the thing, Ben. But I feel like we're slipping off the goalpost here. This is really important. Like you're saying you can't give me one, but I need you to appreciate that if you can't give me a goalpost, you're not operating scientifically. You're not operating as you're not giving me any information of what could change your mind. You're not coming to this conversation with a idea of what could possibly change your mind if you're wrong. And that tells me, like, that's it may not be about the science for you. This may, and I don't want to psychology, but again, if there's nothing that you can tell me that would change your mind, because I can tell you things that would change my mind. If you can't tell me anything that would change your mind, we have a problem.
Yeah, I agree that if someone can't present data that can change someone's mind, that
I'm not asking. No, I'm not asking. I'm not asking you to present data. No. I'm not saying no. That's not what's going on. I'm not asking you to present data that would change your mind. I'm saying, what would the data be? What would that even be? Do you have any idea of what the data could be that, if it were the case, would change your mind? And if you can't give me an answer to that, that's where the problem is.
Okay, so can we set an arbitrary goalpost of 1 in 5,000?
1 in 5,000. Okay, so we can. The problem with that, though, is that would make just about every pharmaceutical not safe on your.
Okay.
So that would just be a reductio.
Okay. So can we move on to the autoimmune data then?
Well, no, you have to appreciate the reductio there. Like, again, so if you can say 1 in 5,000, the problem, the issue with that is, if you say 1 in 5,000, you have taken a view, again, as I said in the beginning, you've taken a view of safety so high that you'll see this with just about anything. You'll see SAEs in just a higher rate of 1 in 5,000. Theoretically, if you increase the sample size high enough, you could probably see that just about anything, Ben. So the question, and that's fine as long as you're consistent. Okay. If you're consistent with that, if you want to bite the bullet on that and say, you know what, just about everything's not safe, we need to look into everything. But then I would wonder why you're talking about vaccines then, because there are things that are much higher than that, and that are being used more widely than vaccines. But anyway, that's a different point. But you have to bite the absurd bullet. There's a bullet you have to bite with, and that's to say that just about everything, as far as pharmaceuticals go.
So where are the goalposts then?
Yeah, I already gave you the goalpost. So my goalpost, yeah. So the goalpost I gave you was, if for SAE specifically, because that's on the topic we are. Number 1, you would need to be so either or one of the following. The data we have either would be statistically significant or doesn't, or if we didn't have the numbers to calculate statistical significance, then the percentage, the absolute percentage difference, if we have a sample size of several hundred individuals or more, if we have a rate of the first I gave 5, but I said we could go down to even 2.5, 2.5% or greater for SAEs. The rate, so for the rate of SAEs of, I'll even go down to 2%.
Sure.
If we have that greater between the Gardasil and the placebo, then yeah, for me that would be like, hey, why are we having that? Especially if it's statistically significant with a population size of several hundred or more, unless it's superseded with the qualification, unless it's superseded by larger studies showing that wasn't like an actual accurate thing. Mm-hmm. So I'm giving you a goalpost. It has numbers, it has specific metrics. So the debilitating SAE specifically, and it has a qualifier. Those are all tangible things I can give you, and that would change my mind if the data turned out that way. So I am operating in good faith here. I'm telling you, here's what the data would be that if you showed me tomorrow this study, it would change my mind. 2%. I just want to have, and notice I don't see any reductios on those goalposts. I think those are reasonable. But so either, all I've heard from you so far is that you either can't give me a goalpost, or you will give me a goalpost, but you'll put it so low that you'll have to bite absurd bullets on everything being.
Okay, so what if my goalpost was 1 in 1,000 or 1 in 100?
1 in 1,000 or 1 in 100. Okay, so 1 in 1,000. So the 1 in 1,000 be biting that same bullet for many, many things, if not all things. Because you do see SAEs for 1 in 1,000 just because that's a rate you see it in, and you do see. If we go through just even Tylenol or NSAIDs, or if we go through even vitamin B, maybe even vitamin B12 injection. But if we go through just about a whole bunch of things, you would have to bite the bullet on that as well. If you go to something like 1 in 500 or 1 in 200, or 1 in or 1 in 1 in 250 or something, around there, you start getting into the territory where it's like, okay, this is starting to become more reasonable. If you go in one, but certainly 1 in 1,000, when it comes to 1 in 1,000, certainly in that case, I would say you'd be biting an absurd bullet. You'd be saying that just about every pharmaceutical, if we're talking about those SAEs, just about every pharmaceutical for you would be not safe on your view, Ben. To me, I would just register that as a bullet that's absurd.
Okay, so can we move on to the autoimmune data then?
Well, I'm going to switch headphones real quick. We can move on to. Hold on, sorry. We can move on, but I don't know if I actually don't know if I can move on, Ben, because when I engage in a scientific discussion, unless you want to just bite the bullet. Look, if you're going to bite the bullet and say, like, you know what, my concern for safety is just so low.
Or high?
Yeah, sorry, it's so high. I'm sorry, I'm just getting sorry, 1 second. You don't need, I have to change my headphones. I'm sorry about this because my battery ran out of
okay, go ahead.
My AirPods. Sorry about that, Ben. Connecting and reconnect for here. Okay, yeah, so just to be clear, I'm not saying you need to know what would convince you of something. It's just that if we give you an example of what would convince you of something and that wouldn't, then there are entailments for that for other cases unless you have symmetry breakers. So if you say, I don't know if 1 in 1,000, if 1 in 1,001 or something, or 1 in 2,000, I don't know if that would convince me. If you say that, then you also would have to say that I don't know it would convince me in other cases unless you have a symmetry breaker. So that there would be entailments on that would be absurd in my view. So if you want to bite the bullet on that, that's fine. I just want to make it clear
that you have a standard of safety that is so high that you are not comfortable with saying that even the most minor of things, as far as pharmaceutical companies would go, that actually are safe. And so long as you're okay biting the bullet on that in this debate, that's fine. We can go on to autoimmune. But just to make it clear to everyone listening that you have you're setting a standard of safety so high that there's really, you'd really be questioning, you really wouldn't be convinced that just about any pharmaceutical would be safe.
I mean, I am still not able to understand how you can say, okay, we have about 1,100, and that is a big enough data set to like know that what we're looking at is.
Yeah, sure. Yeah, I can help with that. So the reason why then is because even if we theoretically at this point, even if we theoretically had 20 million people, even if we theoretically had 20 million people in both groups, and it turned out to be the same, it turned out like there's no way we need more people, we have enough, and it turned out to be that rate. It turned out to be 2 out of 1,100. If you want to say that degree of SAE, which by the way, there was recovery from, no one died in those SAEs. We're not even talking about death. We're talking about an SAE that didn't result in death and they resulted in recoveries. But even if you want to say that is your level of type of SAE that we don't have enough for, the problem is even if we theoretically did have enough, even if we had a magic control group and we just set it all to 0 with millions of people in it, I would say that rate is so low that in compared to other pharmaceuticals that everyone considers safe, like almost everyone considers safe, just about any pharmaceutical, then in order to say that that's not safe, you would still have to say that all these other things are also not safe. So it's, unless you could bring a symmetry break.
What do you mean bring a symmetry?
Yeah, a disanalogy. So for example, if there is a medication that's an injectable or not an injectable or whatever it is, you can say like, okay, my bar for this medication is really, really high. My bar for the other medications are really, really low for safety. But then it looks like you're having an inconsistent standard of safety for these medications. And then we would ask, why are you okay with having, saying something is safe when you have a lot of SAEs in one and not in the other? Why do you have different standards of SAEs with respect to SAEs? So you would have to provide a justification for that for all of the, and you'd have to, in this case, you'd have to do it for all of these other pharmaceuticals.
Sure, fair enough. So let's say we set the bar at 1%. We don't know. Okay, so any theoretical extrapolation of the data we have is a theoretical extrapolation. So
no, but I'm giving you the Steelman, Ben. I'm saying even if we had 1 million people in the control group, even if you're saying the issue is the control, I take it to be the issue is the control group. Because look, I think 1,000 is enough. Like I don't know any power calculation here that's going to say, okay, like over 1,100 people. I don't know what else is being looked for. Now you maybe say, okay, 500 is not enough in the control group. Okay. Theoretically, even if we had 1 million people in the control group and they all had zeros down the line, it would still wouldn't get you to 1%. It would still be 0.016 or whatever.
But you're doing an extrapolation. We don't have.
No, it's not an Ben. It's not an extrapolation. I'm giving you the ultimate Steelman. Because really, if I were to extrapolate, I wouldn't even say that there were zeros. I'm giving you the case where, of all the possible things that can happen, it's not, again, this is not an extrapolation. An extrapolation is I'm trying to approximate what's going to happen. I'm saying of all the possible things that could happen to the control group, I'm just talking about the control group now. Of all the possible things that can happen to the control group, if the greatest Steelman for your case, literally the greatest Steelman that possibly could be, which is we scale it up to 1 million and everyone does magnificently, something that would never happen in reality. If we scale it up and not a single person has 1 SAE, even if we give you the ultimate Steelman, again, not an extrapolation, it's just of all the possible things that can happen in the control group, we give you the greatest case possible. We give your side the greatest case possible. You would say, oh, look, we had millions of people, nothing happened here. Here we have 0.016%. It would still look absurd.
No, the greatest steel man possible would be that you take a bigger sample size and that severe adverse event rate actually goes up.
Wait. In which group?
In the vaccine group.
Yes. Oh, yeah, I agree. Yes, I agree. Okay, so then my question to you is, where is your goalpost? Where is your goalpost for the power analysis then? What's your power analysis to show that this is not significant enough to detect given differences of a given level of expectance? And what sample size are you comfortable with, such that if we did have such a sample size, you would say, okay, I'm okay with saying that this is safe now. What's your goalpost for the sample size?
So in the insert, there's about 20,000. The sample size is about 20,000. So I would like to have a 20,000 sample size.
Okay. And just to be clear, for all the other pharmaceuticals that only had around 1,500 and not 20,000, you're going to say that those aren't safe either. Is that correct? Do I have that right?
That's different. Oh, and I'll explain why. Because this is a drug that's supposed to be given to everyone. Okay? These other drugs are not supposed to be given to that sample size of a population.
Okay, great. Just to be clear, if the law changed, if the recommendations changed, that these are drugs that were not given to everyone. Let's say theoretically tomorrow, the recommendations changed and then they said, oh, we can just give these medications as you want. It's it's up to you. We're not going to make any laws pressuring people or anything like that. Are you now going to say that it is safe?
I'm sorry. Explain that to me again.
Well, yeah, I asked for a symmetry breaker. You provided a symmetry breaker. Your symmetry breaker for safety was the laws or recommendations pressuring people surrounding the drug itself. So you've offered a symmetry breaker, and now I'm just asking if that symmetry breaker wasn't there. So if the symmetry breaker wasn't there, just to be clear, if tomorrow the legislators changed their minds, and then they all said, well,
I'm talking about this drug was designed to be given to everyone.
Great, so great. Say it wasn't. Great, Ben. That's fine. Say it wasn't. Let's say the medication didn't have a design in mind. Let's say the people who designed it didn't have that intention in mind. Are you now going to say that your view has changed and now it is no longer unsafe or unknown to be safe? Now it is safe. Do I have that right?
No, I'm saying that if the
okay, then that's a contradiction. So now you're in a contradiction right now.
No.
So the reason. Yep.
What I'm saying
okay, go ahead.
I would need a larger sample size if this drug is going to be distributed in a wider population.
Say it wasn't. Wait, you actually didn't say that before, but now we can go with that. So let's say let's say it wasn't distributed and designed to be distributed in a wider population. Let's say someone just developed the drug. Here's the drug. My question to you is with respect to injecting it into people. Is it safe? Now, you offered a symmetry breaker for your epistemic standard of 1,000 versus a 20,000. Your symmetry breaker between other pharmaceuticals and this medication was it is designed to be given to a wider population. Now, I can harp on what that means, but I don't need to. My question to you then is, let's say that wasn't the case. Let's say someone develops the Gardasil vaccine, and the Gardasil vaccine did not have the intention of being distributed to the larger population, and then you have the data of 1,500. Are you now going to say that that injection is safe?
That we got basically the same. I'm not sure what you're asking exactly.
Okay, so Ben, your reason you're in a court. So this is a problem for you, Ben, because here's the reason. I can explain it. So look, we're talking about applying epistemic standards consistently. Yes. I asked you, what is your standard? Why is your standard for vaccines different from all the other pharmaceuticals? You gave me an answer. Is that your answer was that the vaccine was intended to be distributed to a larger population. My question to you then is, if that was not something that was true of one but true of the other, if we equalize that out, so if the Gardasil vaccine was developed and it was not intended for wide-scale distribution, if it was just intended the same way other medications or pharmaceuticals were intended, on your view, would you say that it now is safe?
No, that's
a logical contradiction. Yeah. So now I can actually spell out the propositions that form the contradiction.
No, I don't think it's safe. I said my desire for sample size is based on just how many people are receiving the drug. Because
I understand that. Okay. I get it. So there's nothing about that I didn't understand. So your desire, yes. The question was for you to be able to say that it's safe. For your goalpost is higher for drugs that are distributed greater, that had the intention of being distributed greater. You said it's designed for wider distribution. Now I asked you, say it wasn't. Let's just say that the Gardasil vaccine was developed without the intention, that the makers of the Gardasil vaccine did not intend it to be with a widespread distribution in mind. Are you going to now change your view and say that it is safe, that the Gardasil vaccine is safe now?
No, I'm not going to change.
So now you're in a logical contradiction. The propositions that form the contradiction on your view are it is and it is not the case for the Gardasil vaccine that the bar that is being set, it is and is not the case that it is based on the intention of widespread distribution. Because you told me, I asked you, why is it at 20,000 and not 1,500? And you told me it's because it's based with the intention of widespread distribution. Then I asked you, if it was not, would it then be safe? Would it then not be the case that it is the goalpost here is 20,000 and not 1,500? Then you said no.
No.
So the proposition, yeah.
Goalpost, I change the amount of data that's satisfactory for me to be able to make that decision.
Right, based on the symmetry breaker, which is the intention of widespread distribution.
So.
Right.
Okay.
Based on the symmetry breaker, yeah. Yes or no? Based on the symmetry breaker of the intention for widespread distribution.
Yes, the degree to which it's widely distributed changes my goalpost on how big the sample size should be.
Oh, okay. So it's not the intention of widespread distribution. If it is widespread distributed, if it's distributed in a widespread manner.
It's a semantic just runaround. And
well, that's important in propositional logic, and it's important. Yeah. So we can talk. Okay. Well, but it's fine. We can go with that. So then my question to you again, Ben, will just be the same, just modifying the semantics. Let's say that the Gardasil vaccine was not widely distributed. Let's say that it was distributed to the same degree as all the other pharmaceutical drugs, to the whatever degree. It's like the other pharmaceutical drugs on average, to whatever degree that they are on average distributed on, that the Gardasil vaccine is distributed on. Are you then going to say that the Gardasil vaccine has met my goalpost for safety? And I would say, yes, it is safe.
I can't answer that. What I can say is the amount of data that I would need to see.
Ben, come on, man. Look,
you can say, oh,
how can you? What?
But we're not just, like it's. You're basically saying.
Well, look, Ben. This is a contradiction. You're in a contradiction now, Ben. First, you told me that the difference which makes the symmetry, the difference which makes the epistemic standard was X. We can call X the widespread degree. And then I said, if X is not there, would that not make the symmetry breaker? If it's only X, and I say, well, X is not there, then the symmetry breaker shouldn't be there, and your standard should be in accordance with everything else. Now you're saying it's not the case, or now you're saying you don't know. That's a contradiction of what you first said, which that you do know that it's X. So you're still in a contradiction here.
So my argument the entire time is that I don't think the data are sufficient to make a decision.
I understand that. And my question is, well, hold on. That's not the no. That's not the proposition. The proposition was not whether the data is sufficient to make the decision. The proposition was the Gardasil vaccine is safe. Is what we would call safe. Now, The question to you was, if the proposition is the Gardasil vaccine is safe, what is your goalpost for the Gardasil vaccine being safe? You offered a goalpost. What's your goalpost for sample size? You offered a goalpost of 20,000. Then I said, just to be clear, everything that's under that, all the other pharmaceuticals, you're going to say, we don't have sufficient data to say that's safe too. Then you tried to give a symmetry breaker. Your symmetry breaker was, I am okay with the 1,500 if it is, I'm changing it to 20,000 because the widespread distribution. That was your symmetry breaker. Now I asked you, what if your symmetry breaker is not there? Would you then conclude, you know what, the Gardasil vaccine is safe. We have enough data. You saying you're trying to not, you said no, and now you're saying you don't know. Both of those answers are contradictory to what you said the first time around. And if we want, we can go through the propositions that form the contradiction.
Okay. So I guess my assertion all along has been that I don't have enough data to know for sure. That I feel that we need more data. You are trying to pin me down and say you have to have an exact line in the sand of where it's safe and an exact
doesn't have. That's not what I'm doing. I'm not asking you for an exact number. You can give me a range. You can give me a ballpark, as long as that ballpark is not going. So look, first of all, I'm not asking you to. I'm giving you I'm asking for what's enough data for you, a ballpark, for you to say, okay, to whatever reasonable certainty we can say for any of these other pharmaceuticals, to whatever reasonable certain degree we can say for all these other pharmaceuticals is generally safe. Because again, we're just keeping the same epistemic standard for everything unless we have a symmetry breaker. What is your ballpark? You don't have to, it doesn't have to be exact. What is your ballpark number for the sample size required for Gardasil? And I'm just going to compare that to the sample sizes for other medications. Now, you said 20,000. You said 20,000 for Gardasil, but not 20,000 for the other ones. I asked you why. You said because of the widespread distribution. I asked you if the widespread distribution was a trait or was a predicate that was not there of the Gardasil. Would it now change your view on the safety? Would you now have the lower goalpost of sample size? You said no or you don't know. That either means that it's a contradiction, or you would not know for the other cases. You don't have an answer to explain why you have that for the other cases and not the Gardasil case. So I'm just going to go back to the same question.
You're saying that I didn't answer your theoretical proposition where Gardasil wasn't widely distributed.
Correct. Well, you did. Well, you did answer. You answered in answers that either result in logical contradictions, or you answered in well, actually, so far, the two answers you've provided actually both result in logical contradiction.
So I don't understand how that's relevant.
Yeah. So the reason it's relevant. So hypotheticals are very relevant in this conversation because it's important to elicit your epistemic standard of what it is, and if it leads to absurdities or logical contradictions. If you're operating from a place where your epistemic standard either leads to an absurd standard when teased out, or a logical contradiction when teased out, such as absurdities, such as, well, my view on if it is safe or not would actually be influenced. Right now we're not even in the reductio. Right now we're just in logical contradiction. If we can show that your view is a logical contradiction in propositional logic, then we can show that you're, through something called the principle of explosion, even if it's a hypothetical, we can demonstrate that your epistemic standard actually justifies actually affirms all propositions. It's something called the principle of explosion. So right now, both answers you've given me both lead to logical contradictions. Of course, this is assuming classical logic. There are other logics like paraconsistent logic for all you logic nerds that I know I'm aware of that don't explode. But assuming classical logic, the principle of explosion would lead your view to explode on both of the answers you gave me. And by explode, I mean affirm all proposition. That's why it's important that even in these hypotheticals that you maintain logical consistency.
Okay, I don't see how the hypotheticals are relevant. But
yeah, so it's relevant because if in the hypothetical you give me a logical contradiction as an answer on your view, then in all cases on classical logic, you are going to affirm all propositions, whether in the hypothetical or the real world.
Okay, but I mean, this is also based on the idea that it's basically the sample size versus size of distribution is a linear relationship, which I
No, it has nothing. It has nothing to do with that, actually. If you give me a logically contradictory answer in this hypothetical, you're going to, on classical logic, affirm all propositions, regardless of the relationship between sample size, if the sample size is a linear, regardless of the distribution of the sample size. It will affirm all propositions. That's why you need to maintain epistemic consistency. You need to maintain logical consistency in your epistemology. Unless you're going to take some view other than classical logic, unless you're going to say I only affirm paraconsistent logic, I don't affirm classical logic, or if you're going to take some view that you're a dialetheist, that you affirm logical contradictions do exist and are real, and it's not a problem on your view that for logical contradictions to be the case. If you're going to take one of those views, that's fine. But right now you're in a logical contradiction, and you can step back and back off of it and get out of the contradiction. But as far as I'm concerned, when you're in a logical contradiction, that's a problem for your epistemology. And dismissing it as a hypothetical is a very weak response for many reasons. But I think I've elaborated on why that is.
Okay. So can we move on to the whatever point you've made, and we can move on to the autoimmune disease?
Yes. If you're going to bite the bullet and say, listen, I have a logically contradictory position with my epistemology, with my standard is logically inconsistent. I am in a logical contradiction, and I'm okay with that. Then yes, we can move on to the autoimmune stuff if you're willing to concede that point.
I really don't understand your assertion, honestly. So
so then it's important that we don't move on then. So here's the issue. So I asked you in this conversation, the issue was regarding the sample size. I asked you why you require a larger sample size for Gardasil, and you do not require a larger sample size for other pharmaceuticals. You answered me, and you told me that the reason why I require it specifically for Gardasil is because it's distributed more widely. Now, my response to that was, hypothetically, if Gardasil was not distributed more widely, would you be okay with the sample size? Would you not require a larger sample size, just like all the other cases of pharmaceutical companies that don't have it widely distributed? Hypothetically, if it was distributed to the same degree as all these other pharmaceutical companies, would you not have that symmetry breaker in place? And would you say, I don't require a higher sample size? You first said no, that's a logical contradiction. Then you said, I don't know, which is also a logical contradiction.
So I don't think I said no to the proposition. So let's say I say yes. Okay, then you can basically explode it down. It's gonna be
okay. So if you say yes, So if you say yes, there's a reductio there. If you say yes, it means that your view on whether a drug is safe or not is actually dependent on its distribution. It's not dependent on the properties of the drug itself and how it interacts with people's bodies. You're saying that the whether a drug is safe or not actually depends on how many people are taking it. That would be a reductio ad absurdum.
I don't think that.
I think there's a point, Ben. I think there's a point you're trying to make I can try to steelman it for you. I think what you're trying to say here is that because a drug is distributed to so many people, we should have a higher bar for distributing it to that many people. We need to have a higher bar for safety, not to say that it's safe, but to have a higher bar for safety just because there's a precautionary principle that if we're wrong, that would have a greater consequential effect than drugs that aren't distributed as greatly. That is something you can say. However, that is still tangential to the debate proposition of if Gardasil is safe. That is a proposition of deciding what to do, not a proposition of if Gardasil is safe. You can say Gardasil is safe, however, because it's distributed so widely, I want to make sure it's even more safe than everything else. That's something you could say. That would be logically consistent.
Okay. And I've told you from the beginning that my assertion is that I don't feel we have sufficient data to make the decision. So wait.
Wait. I'm not, but the proposition wasn't whether we're making a decision. The proposition was Gardasil is safe. The Gardasil vaccine is safe. So I'm trying to explain to you the difference between those two propositions. Proposition 1 is the Gardasil vaccine is safe. Proposition 2 is, do we have enough data to make a decision to inject Gardasil? Those are two different propositions. One is the debate proposition, one's not the debate proposition. So if you want to say, when you say the standard, so what I'm steelmanning you documenting this for you. If you want to say, what you can say is that, yes, Gardasil is safe based on everything that, based on just as far as any medication that is not widely distributed is safe. However, because Gardasil is widely distributed, I set a higher bar for what the safety, the degree of how safe it is, even though it is safe or is as safe as any other medication that we widely consider to be safe. I have a higher bar for making the decision to distribute it widely because distributing it widely can have consequential effects. That's fine. It's also just tangential to the debate proposition. The debate proposition was, is Gardasil safe? Once you say something like that, then you've already conceded the debate proposition.
Okay, so I have many reasons why I would like to see a larger sample size.
Okay.
And yeah, and so I just don't feel that 2 out of 1,000. Like if I steel man it for you and say 2 out of 1,000 of the severe adverse events, only 2 happened. I don't feel that 2 out of 1,100.
By the way, I don't think that's a steel man from. I don't think that's a steel man for me. The steel man, 2 out of 2 again, because the 2 are still unknowns. Yeah. I think those 3 are like very, very, especially the 2 are very certainly not related to the vaccine. There's hard data for that. But anyway, so let's just hone in on this. What's your symmetry breaker for why you require a higher sample size to say that it is safe? Let's just get that on the table.
Look, I'm not a statistician, okay, and I'll say that right out there. But if there is 1 to 5 out of 1,100, that's enough for me to go, okay, we need to study this more.
Okay, then I asked you, that's going to fall into the category of just about any other medication. So now let's repeat the question. Are you going to also bite the bullet on just about every other medication having that rate for SAEs?
I haven't studied, traditional prescription pharmaceuticals enough to know.
Okay, say I provided you with the data. Say I provided with you data for all of them. Are you going to say all of them are now, we need more data to say anything is safe? Because I understand there's a way out of here, which is to say, well, the symmetry breaker is I haven't read the data. And I almost, by the way, I was thinking of actually, I anticipated this, and I was thinking of actually just sending you safety data for all sorts of medications that people consider safe, just so that pathway is closed off to you in this debate. But I didn't do that to you. Okay. But because I'm assuming you're going in this with good faith. So my question to you is, say I showed you all these medications that everyone considers like, okay, yes, medications can be unsafe to whatever degree, but as far as medications go, this is pretty darn safe. And I show you that there are SAEs in 1 to 5 out of 1,100. Would you then say that all of those medications are also, we need to look into it more if it's unsafe? Or are you going to bite the bullet on that as well?
That's a theoretical, because I don't even know if
dismissing a hypothetical, dismissing it, that's not a valid move here. Dismissing it on a theoretical question, again, it's a question that is theoretical, but it's testing you to see if you're epistemically consistent. That is a fair move for me to make here. You dismissing it is not a fair move. The reason it's not a fair move is because if it is true, if it is to be the case, if it were the case in that it would actually manifest in reality, it is a relevant question. The reason it's not the case, well, really the reason it's not a fair move is because if it demonstrates a logical contradiction, whether it's in a theoretical or whether it's in the real world, you're on classical logic, your position just explodes again. You're in a logical contradiction. So again, I'm giving you the question. The question is being offered to you. If I were to show you data showing that that rate, 1 to 5 out of 1,100 SAEs, and all these other medications that everyone would be like, oh, like as far as medications go, this is for sure safe. But again, like it's safe as far as medications go. Like everything obviously has an issue or can have an issue. But yeah, so if I were to show you that data, are you going to say then, oh, well, these medications also we need to look more? That's the question on the table. And I haven't heard of response other than you saying, well, you're trying to get out of it by dismissing it as a theoretical, which is not a valid move here. All right, I'll let you go.
I just don't see how it's relevant. Obviously, my viewpoint's going to change if they were deaf or they were stubbing a toe or they were, all these factors that this is getting to the point of absurdity.
No, I don't think it is. I think the only thing, everything that I'm bringing to the table is not absurd. It's actually right on point. I think avoiding this question is what's absurd here, Ben. The reason why it's relevant, again.
Avoiding the question of whether or not I want to answer whether Gardasil is safe?
Yeah, so because again, the reason why this is relevant, the reason why it's absurd not to answer the question of the theoretical questions I'm giving you is because if it seems like you're in a position here, then it seems like you're in a position where you have strung up an epistemic standard where what you're trying to do, it seems like what you're trying to do here is try to separate Gardasil from other pharmaceuticals that are widely considered to be safe. You don't want to, it seems like you don't want to bite the bullet on saying all these other pharmaceuticals are not safe, because then you look a little crazy. And everyone's going to see you like, wait a minute, he's saying everything's not safe. That's kind of loony. It also seems like you want to maintain this position that Gardasil is not safe. And so what you're trying to do, and so what you're trying to do, one second, what you're trying to do is you're trying to offer a symmetry breaker. You're trying to offer something disanalogous. Now, I'm just asking you in the hypothetical if that disanalogy wasn't there, if you're willing to bite the bullet. And it seems like you don't want to answer that question because you realize how absurd it would be to bite the bullet based on that difference. But you can go ahead. The relevant point here is that you're in a contradiction. And whether that's in a hypothetical or theoretical or not, if you are in a contradiction on classical logic, your principle explodes on the principle of explosion. You will affirm all propositions.
So I'm basically saying that okay. So you're basically saying that if I don't agree that if other drugs had similar things, and if I didn't agree that they were unsafe, that I would be logically inconsistent.
Or if you didn't affirm that they were safe based on that standard, you would be in a logical contradiction. Yes. If you set a goalpost for one and not the other, and you say the symmetry breaker is X, and I say, well, if it was not X in this hypothetical, would you say that it is? Would you say that the Gardasil is safe? If you're not gonna say yes to that, where you would say yes to the other cases, and you're affirming that as the symmetry breaker, you will be in a logical contradiction. That's correct. And if you're in a logical contradiction, your view affirms all propositions on classical logic.
Okay, I feel like this is just an absurd question to ask, because what constitutes a serious adverse event is a range of things. And so,
we've been through that, Ben.
Yeah,
Ben, we've been through that. I can granulate it. That's fine. So I acknowledge that it's a series of things. But Ben, I'm not asking you again. It's fine. You don't have to give me a number for all adverse events. You can break it down by the severity, the disability. You can granulate it for me. That's okay too. We can do that. So I'm not forcing you into that. That's not true either.
Okay. Well,
the point here that I need you to appreciate, Ben, the point that needs to be appreciated is that when you have offered me a standard for one and not the other, for A, you offer standard X, for B, you offer standard Y. And I ask you, why do you offer A for standard, X for standard for A, and Y for B for that standard? And you give me symmetry breaker Z, and I say, if Z was not there, would you have the same difference in standard? And you say, I don't know. Or you say, yes, I would have the same difference. That's a logical contradiction. And so if you have a logical contradiction, your position explodes on classical logic. That's the problem. You're contradicting yourself. And I think I'm just detecting, I'm not trying to, I don't want to psychologize you or whatnot, but I'm detecting you're getting into these contradictions because you do have different standards and you're not able to address the difference in symmetry breaker in these hypotheticals because, I don't know, are you coming from a motivated place or not? I don't want to get into the psychology. What I want is the symmetry, either a symmetry breaker, which you're willing to bite the bullet on if it's not there. I want you to get out of the contradiction. Or I want you to accept that you're in the contradiction and you're willing to have a logically contradictory position to maintain this view that Gardasil is not safe. Okay. One of those things.
Go ahead.
I
will not assert that it is safe or unsafe. I will not.
I got you. I understand that. I understand that. Okay, well then I can ask the same question for agnosticism. That's fine. I can ask you the same question for I'm not willing to take a position. Are you willing to take a position on just about any other pharmaceutical, Ben? Are you willing to take a position on every other pharmaceutical where someone is going to say, okay, well, everyone, like, look, as far as drugs go, okay, but with all these other drugs, as far as drugs go, they're pretty safe. If you're willing to bite the bullet on saying, well, I'm not willing to take a position on any pharmaceutical. If you have that view, that's biting the bullet. That's a reductio ad absurdum on mine. That would be biting the bullet on absurdity, and that would sound deeply unconvincing. You can do that. That's one option.
You're saying that if I asserted that I don't have an opinion on whether any drug is safe or unsafe?
Yeah, if you are setting your bar for safety so high that you will get to the conclusion that you don't even know if any drug at all you could consider it, safe or unsafe, then that would be a reductio ad absurdum. You can do that. Yeah, you can say Gardasil is a drug where you don't have a view on if it's safe or not safe. But if the way you get there is by raising your bar of safety so high that you're going to say that for just about every pharmaceutical, then that's deeply unconvincing. That it seems like you're just raising your bar just to get Gardasil under the bar. And you're going to have to just bite the bullet on everything else that follows under that bar, as being you can't have a leaning or a strong position on whether it's safe or unsafe. You just you don't have enough to even take a view. And again, we can go through data on a whole bunch of other drugs. I could have sent you PDFs after PDFs, just like that 400 document, all sorts of other drugs, just for this point in this debate, just the logically consistent point. I didn't. So I won't do it in this debate because we agreed not to do it. Okay. But the key here is the point still stands. The conceptual point still stands that what's deeply unconvincing here is if the only way it seems like you're able to get to Gardasil falling under the bar of something that you're not going to affirm or take a reasonably strong position on being safe is if you're raising it so high such that all these other things that everyone is practically considering safe falls under that category. Then it's deeply unconvincing. You can have that position. That would be a logically consistent position. It wouldn't get you into a contradiction. However, it would look deeply absurd. Right now, however, but right now you're not in that position, Ben. Right now you're in a logically inconsistent position. So you can back off of that. You could change your view. Right now you're in a logical contradiction.
That it would be deeply absurd if I said that for all drugs, I don't know whether or not they're safe or unsafe.
In virtue of me setting the bar that high, yes. In virtue of you, if you want to say, I have set the bar this high, and therefore, for just about all drugs, I cannot take a view that any of these drugs are generally speaking safe. If you want to, or safe, like when we say safe, we agreed in this debate that we don't mean perfectly safe. We mean, as far as drugs go, every drug can have some risk, but not all drugs have equal risk. And there are some drugs that, like, generally speaking, they're considered safe. We don't mean safety as a perfection.
And so what we want to do is quantify the risk. Am I correct? Okay.
Before we even do that, what we want is a consistent standard of safety at that level. What we want is a logically consistent standard by which you could say, really what we would want, if you're asking me what I would want, if I wanted to say that one medication I can't say it is generally safe, and then the other medications I can say they're generally safe, I need a standard by which those medications would fall under my bar of safety, but this other medication falls over my bar of safety. That's what I really would need, unless you can give me a symmetry breaker by which I have different bars for different medications. That's fine too, but however, when we got into that, you're in a logical contradiction. So the point is, you're in a contradiction now. There are several ways you can get out of the contradiction. However, I don't see a way that you're going to get out of the contradiction that's not going to result in absurdity, but you can try.
Okay. So basically, my point from the beginning, I'm not going to take a stance on whether or not this drug is safe or any other drug is safe. What I want to do is go through the data together and let the people watching decide for themselves.
Okay, sure. So just to be, but then just to be clear, so you don't take a view on, okay, so is your view that, so first of all, you have a goalpost, your goalpost is 20,000, is that correct?
That's not a safety. I would like to see that much data. Yes.
Okay, and if you did have that much data, and there was no statistically significant difference, would you then say that Gardasil is generally speaking safe?
If I had no statistically significant difference in what?
In adverse events.
In
SAEs, immuno, whatever it is. Like, again, we're just asking for, look, here's the thing. Look, Ben, you've said, you gave me a goalpost of 20,000. You put that on the table.
Okay.
I'm just asking you.
Sample size.
Yeah, sample size. Yeah, you gave me a goalpost for sample size of 20,000.
Okay.
My question to you is that your goalpost for all these other medications?
I don't have an answer for that. I haven't studied these other medications.
Okay. So look, I mean, here's the problem then. That's fine. You could not know. But then if you don't know, then yes, there could be some symmetry breaker you're unaware of that you'd have a different goalpost. That's true. However, the issue here is that it seems to me like the only way you're going to be able to do this, to get to Gardasil, not saying it's unsafe, is you're also going to have to say, hey, listen, all these other pharmaceutical companies, I'm not aware that they're safe either. And if the way you get there is by saying that, that's fine. You can have that as a position. You can say, listen, the symmetry breaker is I'm not aware of all the other medications. That's fine. So let me say if that symmetry breaker wasn't there. Let's say I made you aware of those medications. Let's say we talked about those medications. Let's say I provided you data, which I was even thinking of doing in the beginning of this debate. Let's say I provided you data on all these other medications, and it turns out that they didn't have 20,000. Would you then say that I don't think we can say that those medications are safe? If the symmetry breaker of you not being aware of it wasn't there?
I mean, you're not going to get me to assert that any drug is safe or unsafe.
That's not what I asked. All right, I'll repeat the question. So what I asked was, are you not going to be able to take a stance, or are you? Sorry, what I said was, are you going to say that we can't take the stance that generally speaking this medication is safe? So that's compatible with what you just said.
No, not we.
Or that you. Fine, you.
Okay. Yeah, I'm not going to take a stance that any drug is safe or unsafe.
Okay, so wait. But then here's the problem then. So your goalpost here is now 20,000 for the Gardasil one. But now you're telling me you're not sure if the goalpost is 20,000 for the other ones. If the symmetry breaker and then the symmetry breaker is you're saying you don't know what the other ones are?
No,
Look, you're having it. Look, I'm trying to just get a clear answer. Like I'm trying to say, okay, look, 20,000 is your goalpost for Gardasil for a safety study. Is 20,000 a goalpost for all these at least one other medication? At least like all these other. There's so many medications to pick from. Is that are you having a consistent goalpost? Are you applying it selectively? Here, look, ultimately, I'll just level with you. What I'm trying to tease out here is, are you being selective in your epistemology just to say Gardasil is not safe? And this is what I'm trying to tease out. And it seems like, look, my impression, being honest with you right now, Ben, it seems like you are. So I want to tease that out. I want to say, okay, do you have a standard that you're applying consistently for different medications? Is it 20,000 for Gardasil? You said yes. Is it 20,000 for other medication? Yes or no?
I mean, I haven't studied a lot of these other drugs, so I can't say.
Okay, you know what? Let's do it. Maybe I should have sent you the data on all these other drugs.
So
And then if you said I didn't,
looked at the
and then
data,
if you,
saw something similar where it was happening, between 1 and 5 in 1100, then I would say, whoa, we probably need more data.
Okay. So then on my view, again, so then in my view, that's a reductio ad absurdum. So you would actually have to say with that, if you bite the bullet on that, you're going to be biting the bullet on a whole litany of other drugs that are widely considered to be generally safe. Now, you can have that position if you want. That's a consistent position. You could say, okay, well, if we see 5, and then we're not really picking a whole lot up in the control group, but we are picking 5 of these SAEs, even though at least 2, if not 3 of them, turned out to not be even related. If we have a percentage of 0.016% of SAEs that did not result in death, that did not result in long-term debilitation, where people even recovered, that's enough for me to say we need more data. If you're willing to say that, that's fine. That's a consistent position. However, that is going to result in a strong bullet for you to bite. And you're going to have to say that a whole bunch of other medications are also things we need to look more into on before we can say they're generally safe, which a whole bunch of people are going to disagree. Okay.
So the autoimmune rate in the other studies that were not placebo is a concern for me that also makes me want more data specifically.
Okay, we can get to that. We just so we're clear on that. But right, we'll get there. But we're talking about SAEs right now. So just to be clear, you can bite the bullet on the SAEs and say, okay, with respect to SAEs, if we see 0.016% of SAEs that did not result in death in the treatment group, that does not result in long-term debilitating conditions, that people even recovered from. If we see that in other medications, then we cannot say, until we have more data, that they're generally considered safe. If you want to say it, that's fine. That's a logically consistent position. I also consider that absurd. And so would a whole lot of other people, but that's fine. If you want to bite the bullet on that, totally fine.
So the 0.016, which I don't agree with you on that number. If it's
what do you agree? Wait. What do you disagree with me on? That number?
So if I do the math, okay, and we say like, okay.
For SAEs specifically, Ben. Yeah. So what do you disagree with me on this number for? What's the disagreement?
Hold on. 1 out of 1100.
Because we already went through the SAEs and we excluded the ones that were clearly not related to the vaccine. Because we looked into what actually happened. Okay. I got 2 that are still that are possible candidates. 2 out of 1179 is 0 times 100. That would be 0.16%.
Okay.
0.16.
Okay,
Yeah.
you were saying 0.016. So okay.
Sorry. Okay, yeah, sure. 0.16. Then I wasn't misspeaking. I misspoke then. Zero.
Okay.
If you want to say for SAE that did not result in death, that we saw 0.16% rate for SAEs that did not result in death, that did not result in long-term debilitation, that did not result in permanence of the SAE that was recovered from, is enough to say that this medication cannot be said to be generally safe. That's a consistent position. You're also going to have to say that about a whole litany of other medications that virtually everyone wants to say they're safe. So that's going to be a reductio ad absurdum. That's going to be an absurd position. You can have that position.
That data point, okay, the 0.16 combined with the data regarding autoimmunity that is in the non-saline placebo.
Okay. Okay, so you're not going to say, okay, that's fine. We can go into the autoimmunity. So it's fine. So if you want to say it's not just because of the SAEs, it's the SAEs in combination with autoimmune. Okay, so we're not saying, so you're backing off on the point. So we're not saying that it's because of the SAEs that we're not considering it's generally safe. That's not what it is. It's SAEs plus something else. So you have backed off the point. So you're taking back the point that we cannot conclude that it's not generally safe because of the SAEs. So we're backing off of that point, and now we're going to try to form another attack on Gardasil, which is the SAEs plus something else. Okay, go ahead.
are enough to warrant doing more research.
Okay, go ahead. So we've backed off the SAE point. Now we're going to try to form another attack with SAEs and combine with something else. Okay, cool. So what's the okay? So what's the point about the because I've done the data every which way on the autoimmune conditions, but like I want to hear your point first. Go ahead.
Okay. So I'm just pulling up page 8 of the package insert, and it basically shows that you know, in this 20,000 sample size.
Wait, hold on. I want to follow along with you. Sorry. I'm just.
Go ahead.
Yeah. Okay. Page 8. Okay, I see it. Yep. Arthralgia, arthritis. Okay. Yeah, I see it. I see them. Mm-hmm.
Okay. So in this particular data set, we show a rate of autoimmunity in both the Gardasil and the saline control plus AHS, right? Which is a consolidated data set. And if we break that out and we look at like what percentage of that is aluminum control versus saline, let's see if we look at the numbers. I think it's like, I don't want to do any degree of certainty, but it's 320 plus 274. Okay. So it's 594 out of 941.
I'm sorry. What's the percentage? What are you trying to calculate right now, Ben?
I'm trying to calculate on page 8. There's the table where saline placebo is consolidated in with AHS, right?
Yeah.
So I am trying to tease out. I'm trying to understand the differentiator. Trying to understand what percentage of that is saline placebo versus what percentage of it is the aluminum control. And so, yeah.
Yeah, well, I can help you with that. Have you already done this?
Yeah. So
have you done this yourself or
3%?
Wait, hold on. So arthralgias, new medical conditions. Okay. Yeah. So I've seen conflicting data, whether it's 7 or 9 for the arthralgias, whatever. We can go with 7. When you sum it all up together and you look at the non-aluminum, you get a 1.2% for non-aluminum, and for aluminum, sorry, 1 second. 1.2 versus 2.1. So there's a difference of less than 1%. And I did a test for statistical significance because they both were able to pick up on the total autoimmune conditions. And there was not only was there no statistical significance, it wasn't even close between the non-aluminum and aluminum adjuvant. So if you want to say that on this basis, when we have all these listed autoimmune conditions, if you say the combination of the SAEs at 0.16% plus a difference in the non-aluminum, a difference in the placebo versus the control versus the placebo versus the medication, which is less than 1%, not statistically significant, not even close to being statistically significant, you would have to bite the bullet on that being true for a whole litany of other medications as well in order to be consistent, unless you provided symmetry breaker. I would also, go ahead.
I don't see any data on the people with the saline placebo contracting an autoimmune condition.
Oh yeah, so it's the same. Yeah, the arthralgias. It's based on the arthralgias. So they included to make it apples to apples. So for example, do you see so arthralgias can be listed they listed it under musculoskeletal, but there are autoimmune conditions that can be listed under different multiple organ systems. It's just a classification difference. So, for example, if someone has joint pain, in the case of over here, they listed arthralgias as part of autoimmune conditions.
Over here, where you at?
Yeah, in the package insert. Do you see in the So you see in the package insert. Let me just find it for you. In the package insert, do you see where it says arthralgia? They didn't just do arthritis or arthropathy. They lumped in the most common one, presumably, in there. That's how they jacked up most of their numbers, by the way, for the autoimmune conditions. That's why the numbers are so high. If they didn't put arthralgias in there, the numbers would be far lower than 2.3%. You wouldn't see it.
It would be half.
Half? I don't know if you could say it would be half. Well, let's see, actually. Hold on, I can tease that
45 and 98 out of 218, right?
Oh yeah. But yeah, it wouldn't be. Yeah, the majority came from arthralgias. So which is fine. Look, again, I'm not going to pick on that. That's fine. You
Okay.
can say I'm willing to put that in because it's true. Arthralgias are it seems to be all. I mean, I can nitpick on other things as well because there's a lot of things that are like, okay, well, they're lumping things in, and they lump things in the non-placebo control group that really do look like autoimmune conditions, like skin conditions, but they didn't specify what they were, so they could have been acne and not autoinflammatory conditions like eczema and psoriasis. I can nitpick, but I don't need to.
Okay.
So let's accept arthralgias as autoimmune conditions. Arthralgias can be listed several ways. It doesn't matter. I'm willing to accept it as autoimmune conditions each way. Arthralgias can be listed. They're part of the musculoskeletal system. It's joint pain. Joints are part of the musculoskeletal system, but they can also be listed as part of the immune system in an adverse event such as arthralgias, because that's a very common symptom to get for certain autoimmune conditions. So, in the saline placebo, when saline placebo alone, I've heard conflicting reports depending on, so in this packet that I gave you, the several hundred-page packet, there were 7 cases of arthralgia in the saline placebo.
Where's that? What page? So I can just go to
Yeah,
it?
sure. It is page 329.
Oh.
Table 245.
Okay. Yep, I see it.
Yep. So you see they just classified it, musculoskeletal and connective tissue. And then they listed arthralgia. Arthralgia it was in the Gardasil group. It was really what you should be doing, by the way, is you should be comparing within the trial because there's intra-trial variability. So in the Gardasil group for arthralgia, it was 1.3%, and in the placebo group, it was 1.2%. And remember, this is the non-aluminum placebo group.
Yeah, we know.
Yeah. So this is the autoimmune condition that was able to be picked up, which makes sense, by the way, because that's the most common autoimmune condition. So you would expect with the lower sample size that this is what would still be able to be picked up in reasonable numbers, 1.3%, 1.2%.
Yeah, and arthralgia is more of a symptom than an actual condition, if I'm not mistaken, right?
Well, okay, so this is what I said earlier. So you can nitpick on that, that's fine. But if you want to nitpick that, then I'm going to nitpick the original case, and that's going to bring the overall immune conditions down as well.
Okay.
So that's why I chose to include it, because again, you can't have the anti-vax cake and eat it too. That's the key here. You have to be consistent. If you're going to nitpick on it being a symptom, you have to nitpick on it for the case blowing up the numbers as well. You would cut the numbers down significantly if you took out the arthralgias from the autoimmune conditions in the packages. So, and again, the other thing I can nitpick on here is that in skin conditions, a lot of skin conditions, especially in the age group of 9 to 15-year-olds in men and women, a lot of them are autoinflammatory skin conditions, but they didn't specify them further. But they did in the other protocols, such as psoriasis or eczema. Those are autoinflammatory skin conditions, which you were able to pick up skin conditions here. Unfortunately, they didn't specify them to a degree of granularity, but they were able to pick up that as well. Interestingly enough, there were more skin conditions in the placebo group than there were in the Gardasil group by percentage. 3.4 in the placebo group, 2.4 in the Gardasil group, whatever that's worth.
I'm seeing the
table 246.
Two. Okay, I see what you're saying. Mm-hmm.
Yeah, they're actually, yeah. And so for various, in fact, this is true for various different conditions. You actually see a little bit of a higher rate of, so you see either, you can see actually a higher rate in the placebo group, ironically, than the Gardasil group for some conditions. So arthralgias were picked up. And so even if we like give the Steelman, even if I give you all the skin conditions, even if I say, let's just say hypothetically, let's give you the ultimate Steelman and say none of the skin conditions, were autoimmune or autoinflammatory. Let's just say it's at 0. Again, I'm giving, of all the possibilities, let's just give you the Steelman. Tallying up the autoimmune symptoms or autoimmune conditions, we still don't get anywhere close to a statistically significant difference between the aluminum and non-aluminum rates of autoimmune conditions by percentage. Now, and even if I were to include the skin conditions, and if I were to write the authors, by the way, and say, well, how many were psoriasis, how many were eczema and whatnot, that were included in the package insert, it would be even worse for your position. The rates would be even closer, and it would be even more far off. Even giving you the ultimate Steelman, it's a complete wash. You just don't see anything different between the placebo and the non-placebo.
On skin conditions?
No. On total autoimmune conditions. Look, in the case of 1,128, the only autoimmune conditions we could even say that they possibly got. So remember, these are 1,128 people who experienced out of that 1,128, the number of people who experienced arthralgias were 15, so 1.3%. And so we were able to pick that up. And the other potential source of autoinflammatory conditions were skin conditions that are not otherwise specified.
Yeah.
All the other conditions were not present in the package insert. So they didn't find anyone, none of the people who had Gardasil developed hypothyroidism, none of the people who had Gardasil in protocol 18 had hyperthyroidism. None of them had Stevens-Johnson syndrome. None of them had erythema nodosum. None of them had those other conditions, which makes sense. When you jack it up to 20,000, there's some amount that you'll pick up. However, some autoimmune conditions, not surprisingly, were picked up. Some or conditions that were considered autoimmune in the package insert. So we had a study, 562 in the placebo, 1,128 in the Gardasil group. The rate of arthralgia, which was picked up, was 1.3 in the Gardasil group and 1.2 in the placebo group, non-aluminum. A 0.1% difference. Completely not statistically significant. I think the p-value for that was like 0.49 or something. It's like literally what you would expect. There's absolutely no difference.
Okay.
And then the skin conditions, which could be autoinflammatory, you actually saw more of them. You saw more skin conditions by percentage in the placebo group than the Gardasil group, by over 1%. 4.7% in the placebo group and only 3.9% in the Gardasil group. So this whole idea that when you look that we're seeing these autoinflammatory autoimmune conditions, we're really not. And you can try to say, well, we need more people, we need more data, but then I'm just going to ask you the same question. We're going to go back into the hypothetical and going to go back into offering a symmetry breaker between the 0.16 SAE and the complete wash here for the autoinflammatory conditions, unless you can provide me a reason.
Sure. Good assertion. So as I mentioned before, What's seen on the insert with the Gardasil versus the AHS is very concerning. Okay. And so that's enough for me to say, okay, if we're testing the aluminum control in this big of a sample size, we should be testing the saline placebo in this big of a sample size too. That essentially one protocol out of 18 is not convincing enough for me.
Okay. So if I can show you, So if I can show you a 2.3% rate of all of these conditions, of the summation of all of these conditions in other pharmaceuticals, okay, would you then say that you require the same epistemic standard? 2.3 or higher.
That for a 2.3 or higher rate, that we need a 20,000.
2.3%. Yep. Which, by the way, just to put this in perspective for you, like, this is not any different. By the way, this is not any different than what you would normally expect to see in the normal population. Like, it's just when you combine things like arthralgia, which is very, very good. The incident, looking at these numbers, honestly, like, and knowing the prevalence of these conditions, like the summation of this, I have a completely different take. I actually think it's shocking how low it is. It's not like it's just concerning. It's actually shocking. When you combine and summate all that, it's actually shocking to me. I was surprised to see how low those things were, given the normal prevalence of arthralgia, arthritis, arthropathy, even in that age group. I was surprised to see 2.3%. It rings of safety to me. But anyway, for your sake,
let's explode that out, because that's an interesting assertion. Because if you're saying that over the course of say, what is it? It's 6.5 months that was looked at, that if 2.3% of people developed an autoimmune condition in that time, you explode that out. What is it, like 50 times 6 months is 25. Within that time, everybody's going to get an autoimmune.
So the key is but here's the thing. Ben. The reason you're getting these numbers is because it was an all-inclusive set, including things that really, they didn't take a conservative estimate. They took the most liberal estimate with it by including things that are symptoms of autoimmune conditions that aren't really necessarily autoimmune conditions. And again, you see that by about half of it coming from arthralgias alone.
Okay. So let's say
the reason. So that's So again, also like psoriasis is very common. Let's see. Where are the other things that are coming from that are common? Again, hypothyroidism. That's also fairly common. But yeah, like arthralgias, arthropathies, erythema nodosum. That's I'm actually surprised to see that's low compared to what it was. IBD. I see. Pigmentation disorder, like they didn't even specify it. Yeah, I mean, this is, Oh yeah, the other thing you see 13 and 15 for psoriasis. These are also other common conditions. So again, like none of this is actually really surprising to me, because when they say autoimmune conditions, that's like people think about, they're like, wow, autoimmune conditions. But when you actually look at where the numbers are coming from, a huge chunk is coming from just conditions that are very, very common, and people wouldn't look at that and be like, "Oh my gosh, I have autoimmune condition." Like, people like having joint pain I have joint pain sometimes. Like, okay, I have psoriasis. Like, that's common. So the numbers are not going to be 2.3%. They would probably be, if you really just took the serious autoimmune conditions and looked at those, they would be significantly lower. And I don't think they would be any more greater than the prevalence in the general population for those age groups.
I disagree with you here. I think that this is, with like time-wise, I'm losing the word, but I'm saying like basically within a certain window of injection, this rate is really high, and it may be high in the general population, but like, that's
no, it's a per year. No. Rates per year, Ben. I'm talking about rates per year. Incidents per year. within different age groups. I'm baking that into the cake already.
Okay.
I'm accounting for what you're saying. Yeah, rates per year. I mean, again, unfortunately, I didn't send you data for this, so I can't use it in the debate, but rates per year, you can look at things like arthralgia for rates per year. They're strikingly high. You can look at rates per year for developing hyperthyroidism. You can look at I'm baking this all into the cake. This isn't anything surprising. It really isn't. I regret not going into this more and spending more time going into the rates compared to the baseline population, because I don't know anyone who would look at this data and be like, oh my gosh, this is surprising. It's really not. But aside from that, the key point here, Ben, is that I'm still going to pin you to logical consistency. I'm going to say, okay, then what if we saw 2.3 when we combine arthralgia, autoimmune thyroiditis, celiac disease, type 1 diabetes, erythematosus, hypothyroidism, etc., combining summating it all together, say in 1 year, we saw 2.3%, would you still say we need to look into it more at that point?
I mean,
because you're going to be able to see that. I'm going to tell you right now, there are many conditions. There are many medications that are generally considered safe that we will be able to see that in. The reason is because these aren't surprising rates, and they're not different from the general population by any significant degree. They're not.
But
if you want, we can pick up the debate when we have all of that data collected, and I can show you that these aren't different from the general population. We can do that too.
Sure, but keep in mind that these are happening in children, not happening. No,
by age group. I already baked that into the cake. I said by age range. I accounted for that already.
Okay, so you're saying that there are 9-year-olds with hypothyroidism? Yes,
at 0.3% per year. There, you can see 9. Yes, pediatricians do have. Pediatricians deal with hypothyroidism in 9-year-olds. Absolutely, 9 to 15-year-olds. I'm in clinical trials. I just saw a pediatric patient within that age range. Not just, but I have experience. I've seen a pediatric patient in that age range with hypothyroidism. Sure, I have, absolutely. You see it. It's not uncommon.
Okay.
These things aren't.
Go ahead.
That's
an anecdotal point.
I understand it's an anecdotal point, but I don't need the anecdote. I'm not relying on the anecdote to make the case. We can provide data to give the case. Like, that's fine. It's just that I didn't send it to you beforehand, so I can't use it in this debate by the debate rules. But you can look into the prevalence of the general population. You're not going to see really anything surprising here.
Okay, but we're showing that these began within a certain allotted time from injection, 14 days, correct?
No. These are new medical. No, that's not true. So these are new medical conditions that developed through month 12 follow-up, not within 14 days.
Okay. So we don't even know how many developed later than that, correct?
We could probably later than that? No, we do. Because what we can do is they did a safety follow-up for these protocols. Most of these protocols had long-term safety follow-ups. And we can get into that data as well. I know the data for protocol 018, they went out to 5 years, even 8 years. And the only thing they saw, by the way, it's interesting. The only SAE that they ended up seeing, they saw one SAE 8 years, up to 8 years later in protocol 018. And that was from a person who had a temporary temporary palsy of the facial nerve. And that person was originally in the non-aluminum placebo. Now, they could have had a vaccine after that, sure. However, they probably did have a vaccine after that. However, just for whatever it's worth, they were originally in the placebo group, non-aluminum. Other than that, in 8 years, no SAEs. Now, I can even call the authors or find the supplementary data for the autoimmune conditions or new medical conditions broken down after that, and we can compare that to the general population. Fine, that's fine. We can do that at a later debate. At this point, though, because these require a lot of work, and based on everything I'm saying, my knowledge of the prevalence, this is a complete wash. I really want to know what would change your mind, Ben. What would change your mind? What do I have to, let me, honestly, Ben, What do I have to find that you would be willing to change your mind on for this? What data needs to bring? So let's say I do all the work for you. Let's say I phone the authors. Let's say I call the author, say, hey, you did a long-term extension, 8 years, 10 years, 5 years, whatever. What were your what were the immune conditions? And then we go to the general prevalence of the general population. What data do I need to find that you would change your mind on? What is it?
So I would want to see rates of autoimmunity in people of these ages and date it back to when the trials were happening. Because as we know, autoimmunity continues to increase. And that, prior to like 1990, 1995, I mean, it was, considerably less.
Well, just one point about that. Yeah, so chronic, the incidence of chronic conditions increasing has been attributed to, you know what that's attributed to, right?
I mean, I've heard many theories, so why don't you tell me yours?
What theories? Well, tell me what you've heard. I just want to know if we're on the same page here. So you may have heard. Go ahead. Or you may have heard like diagnosis, that they're diagnosing more of it. That's one theory that you may have heard. Have you heard any others?
Obviously, like why it's increasing.
Yeah.
Like what specific conditions? Autoimmunity?
No, chronic conditions in general. Like overall chronic conditions that are increasing in general. Like what do you think is supposed to be the reason for that?
Obviously, when it comes to heart disease, type 2 diabetes, we know diet plays a huge role. We've seen an explosion in the vaccine schedule since the 80s when the law was passed that the manufacturers no longer had liability. So now we're at something like 70-something shots, whereas when I was a kid, I'm not sure how many I got, but I was born in 1981, before that law took place and before a lot of these new drugs were brought to market. So we've seen, so that has been proposed as a cause, that the vaccine schedule increased dramatically, and that, glyphosate has been proposed as a cause. A slew of environmental toxins from, so many different industries that have been proposed. There's a lot of things out there that are proposed.
Okay, so none of those are the major, like, the hypothesis for why chronic conditions, including autoimmune conditions, may be increasing. Advanced technology in terms of delivering, in terms of that infants with pre-existing conditions or conditions that would make them less likely to survive would either die in utero. Yeah, go ahead. Sorry. I'm sorry. Are you trying to say something? You're cutting off for me.
You're cutting out. I can't get all of it.
Oh, okay. Hold on. Let me just leave and rejoin the.
Okay.
Okay. Is this good for you now? Can you hear me now?
Still kind of dicey.
Let me try doing it on my computer. Let me try rejoining there. Sorry for a technical difficulty. Yeah. The prevailing hypothesis is actually that it's actually coming from our increase in care for infant mortality with regard to that. So it used to be that infants who had conditions, fetuses who had conditions in utero, would either die in utero or they would have more complications during post-birth, and they would end up dying, and you wouldn't end up seeing those children making their way through childhood. But because of our increased medical technology, we're able to save a lot more of these infants, and our infant mortality rate over time has also dropped accordingly. And with that, which is a great thing, we're able to save a lot of these infants. However, there's a cost to that. Because that we're able to save a lot of these infants who are in worse shape to begin with, those are the infants that are more likely to develop chronic conditions. And that's why we're seeing an increase in the chronic conditions. That's the prevailing. So none of the, so I actually don't agree with any of the almost maybe diet, but I don't agree with glyphosate. I don't agree with like the main thing I agree with is it's just simply due to our we're just saving more infants that would have died otherwise. They have problems. We're able to get our infant mortality down. Our infant mortality drops, chronic disease increase.
Okay. But that would see now we have to basically discuss data that we haven't provided before.
Which is fine. We can pick that up. So again, like just so for consistency's sake,
to me compared to other countries. So
okay, yes, I'm not saying. Listen, I'm not saying that's the only. I'm not saying it's the only factor. We can look at it. Also, the other thing to tease out, we can get into that. Like it's the way our mortality rate, our infant mortality rate, is actually calculated differently than other countries. That by and large, what explains our difference in infant mortality rate compared to other countries. People think we have the United States has a higher infant mortality rate than other countries. It's actually not really true. At the way it's calculated, it's actually in the way it's reported. We only spiked up to the rate we do have now compared to other countries after there was a policy change that resulted in a change in the way the infant mortality rate was calculated. That's actually why we're saying we have a high rate of mortality compared to other countries. We can get into that as well. It's actually a misnomer. A lot of people think we have a higher infant mortality rate than other countries. It's very misleading when they say that.
Okay.
But regardless. So look, Ben, at the end of the day, I don't know. Do you want to, I'll just leave it up to you. So my position here is, if you want to say it's in combination of the autoimmune conditions you see, I'm going to say, okay, I'm going to hold you to that standard, and I'm going to apply that to the other drugs as well. I'm just going to do the same thing I did when you talked about the SAEs. I'm going to say, in conjunction with the SAEs, and autoimmune conditions, I will hold you to that standard for all these other pharmaceuticals. Fine. If you say no, I'll ask for a symmetry breaker. And I'm going to ask you, what if the symmetry breaker wasn't there? And if you say you don't know or no, then I'm going to say you're in a logical contradiction. If you say yes, I think I'll be able to provide you with a reductio because I think this will apply to a whole bunch of other medication.
Okay, so you're basically asking me, and I want to make sure I understand your question before I answer it.
Yeah.
Is that if the rate of autoimmunity from these drugs was 2.3%?
Well, not from these drugs, associated with. We don't know if they're caused by it. They're AEs. AEs are regardless of.
Yeah.
So if it was 2.3% associated with it, yep.
Yes. And would I make that drug as unsafe?
No, that's not the question. I would say, would you say that we cannot say that the drug is generally safe?
Would I? So, okay. So you're asking if I would say that we can't say the drug is safe based on this 2.3% of autoimmunity?
Yeah, for all these other medications.
That if they also showed a 2.3%
by the same criteria, yeah, by the same criteria. So, for example, remember, we're applying everything equal. So we get to this 2.3% by adding up arthralgias, arthritis, arthropathy, autoimmune thyroiditis, celiac disease, diabetes, type 1, erythematodeism, hypothyroidism, inflammatory bowel disease, multiple sclerosis, nephritis, optic neuritis, pigmentation disorders, psoriasis, Raynaud's, rheumatoid arthritis, scleroderma or morphia, Stevens-Johnson syndrome, systemic lupus erythematosus, or uveitis. We add all those up, tally them up, look at all the other medications, and if we see 2.3% or higher, you would say we cannot say that they are generally considered safe, just to be clear.
I guess I could agree to that, yeah.
Okay, so I would say to that, what I would say to that is I think you're going to say just about everything we can't say is generally safe with that standard.
Okay.
And we can get into data. Yep, we can get into data for that at a later time. That's fine. We didn't provide that earlier. That's great. I think that's going to be a huge reductio. That's essentially getting to the point where you can only say Gardasil you can't say it's safe. By the same standard, you would have to say that we can't say just about anything is safe.
Okay. I haven't seen, nothing else has come up on my radar showing this rate of autoimmunity coincidental with,
okay, sure. But just to be clear, if provided, you'll agree to it, just to get the goalpost down.
That if I saw the same thing for another drug, I would assert that we can't consider it.
Yeah,
So,
and just to be clear, if I show you drug after drug after drug, which everyone's saying like, wow, this really is safe. Why isn't this even like being talked about? Of course it's generally safe. If I show you that basically every drug has this, which basically I could if that's the rate in the general population. But yes, if every drug is going to be associated with this, you're going to say I guess just every drug we can't say it's safe.
Any drugs that fit this criteria,
Yeah,
that I agreed to for Gardasil, I would also say we can't say that it's safe.
Okay. So what I would say to that is I think you're going to say that you're going to be in a position with that where you're going to be that there really isn't a drug that can generally be considered safe.
Uh-oh, losing you again.
Oh, sorry. Yeah. What I'm saying is that you're going to be in a position where virtually no drug is going to be considered where you would be able to say it's safe on your view, if that's your standard. And the reason is very simple. The reason is just because every study that really did look for all of these things in conjunction would find this or greater if it is the prevalence, which we can just show if that's the prevalence, and we can make the induction. I mean, we can make the inference. Okay. Do you want, because we spent a lot of time on the outcome-based data. I have read the mechanistic data on aluminum. I've read the response to the original paper, to the original aluminum pharmacokinetic study. Personally, it's like, generally speaking, I'll tell you where I come from here. Where I come from over here is just that for me, I don't see a whole bunch of point to talking about 2 rabbits when we have randomized controlled trials on 1,500 humans. It almost seems absurd. I can do it. I can get into the mechanistic data and why I think that paper is completely wrong. That's criticizing the original paper. I also think the original paper is wrong on some things.
Yeah.
We can, if we want, we can get into it. It just, for me, my personal, I just see I think it's kind of ridiculous. For me, it's like it's a done deal. Just looking at this data. But if you want, we can go into the mechanistic.
What's a done deal?
Oh, the done deal. For me, like, just looking at the randomized controlled trials here, for me, that's like all I need. For me, like, that's all I need to be like, okay, yeah. It's either you're going to conclude that this is generally safe, or you're going to conclude that virtually all these other medications are not generally safe, based on the data I'm seeing here. But however, I can entertain. So for me, the going into the mechanistic data, for me, I can do it. It's fine. I can do it for the sake of entertainment. It's just like humoring the mechanistic, because there's an evidence hierarchy.
Yeah.
Mechanistic data is at the bottom of the evidence hierarchy.
I agree. I think that, we could look at, you gave me some great polysorbate 80 studies, which were, actually pretty fascinating to read. And the aluminum study, again, my assertion on basically both of these is that they don't really tell me what I need to know. That the randomized controlled trial is a much better, gives us a much better data set to look at than going down these reductionist rabbit hole on ingredients. So I guess
so we can here's the thing for me. I'll just tell you my thing for the mechanistic thing. Like, the mechanistic thing for me, like, I've read the paper criticizing the mechanistic study on the rabbits, and none of the criticisms actually get to the point of the inference where you would think aluminum is safe. The inference is just clearly that the reason it's not, the inference is, look, when you actually do the tracer experiment, you inject aluminum into the muscle. The tracer experiment shows you that the amount of aluminum, that the peak level of aluminum that you experience, that is experienced, and I know it's animal studies, but even just assuming the same blood volume, extrapolating the blood volume, just doing the amount injection divided by the liters, the highest peak you get would be less than 1% of the normal aluminum concentration that's in your blood at every given moment. Mm-hmm. And that is just like
28 days. Is that what it is?
Yeah, the 28-day one.
Okay. So,
Mhm.
and I guess we're going to talk about it because it's interesting to me. So can we? Are you cool with discussing the study?
Yeah, we can talk about it because I've read the paper. I've read the paper criticizing it, and I don't think any of the criticisms actually land for what the inference actually is. Like, there's many criticisms of it, but none of them actually get to the point. But go ahead.
Sure. So this is essentially a pharmacokinetics question, right? That it's like, what happened to this aluminum? And so for people who are reading, we did an intramuscular injection on rabbits. We don't have a human study, which is like, I wish we did, right? And we show after 28 days that like it went down significantly. And so this becomes a question of essentially accounting, right? Whereby we show that it's not in the blood and it's not in the urine. It's a metal. Where did it go? Are we autopsying these rabbits and like trying to find it anywhere else in their body? Because we know they're not urinating it out. We know it's not in their blood. Are they pooping it out? Are they vomiting it out?
Well, the key issue is that when you inject aluminum, there's a granuloma that forms around the injection site in the muscle. Mm-hmm. The granuloma is the key here, which is what I wish that the authors really did do. Which would, like, again, this is all mechanistic, so I'm just humoring the conversation, but
sure.
The key to do would be to dissect the muscle at the ejection site and find the granuloma. Because then what you would find there is you would find the remaining aluminum, which would be encased in the granuloma. The key point here is in theory,
because we haven't done it. Okay, so
yeah, but regardless, even without that though, Ben, even without that, I don't even need that. That would just be the cherry on top. The point is that in order to get to all the other body tissues, you don't teleport from the muscle. You don't teleport from the muscle to the body tissue. You have to get there through the blood anyway.
Yeah.
It goes into the blood.
Losing you again.
From the blood. Even if all of it did end up, even if all of that did end up not in the urine, even if it did end up in which it didn't, like they did excrete decent amounts of it. But the point is, in order to get into those other organs, you have to see it in the blood first. It has to be in the blood first.
Yeah.
And so that's why they monitored the blood. The point is that the highest peak that you were able to experience, that was able to be experienced by these subjects, if you just looked at the volume for humans, it wouldn't even be 1%. We wouldn't even expect that to be 1% of the peak. 1% of a difference at highest of the normal aluminum concentration. Now, that plays into where the aluminum went, because the area under the curve would also, if that's true, then the area under the curve of aluminum that made it to the blood also wouldn't be more than 1%. The reason I think these are safe is because they're released so slowly. We know a granuloma form around the aluminum. We know it releases so slowly that the amount that if you actually had a detector continuously, I don't even think you'd be able to see normal variation. That's why we're only doing tracers, because we're separating that from the whole litany of normal aluminum that's in the body compared to this small amount that ever gets into the blood when you do the vaccine.
Okay.
That's the issue.
Okay. I really need to. There's many theories about where it could go.
I understand that, but it has to get there through the blood. That's the key. Wherever it could go, it has to appear in the blood first. It's not going to teleport from the granuloma to the bones. I know they criticize that the bones weren't there, but again, the point is it doesn't teleport from the muscle to the bone. It goes into the blood first, and the blood stream gets to the bone, as it does with the brain, as it does with other organ systems. That's why since everything is channeled through the blood, the inference is made through what we're seeing in the blood.
Okay.
So that doesn't respond to the inference.
But okay, so without an autopsy, we just don't know. Is my position.
No, it's not that. Okay, well, look, if your standard of skepticism is that high, sure. But again, the point is that yes, if your standard of what we can know is that high, sure. But again, the point here is that the inference is valid. The inference can be made to be lean in a strong direction one way or another. If you're seeing in the blood, changes that wouldn't even amount to 1% of what the normal aluminum level is, then yeah, theoretically, I mean, you could speculate anything, but the most reasonable conclusion is this is not going to be consequential because the area under the curve isn't either. And they did do an autopsy. It was just an imperfect autopsy. They didn't do an autopsy on the femur. Oh my gosh, all the anti-vax papers go crazy. There wasn't on the femur. Okay, the core inference isn't even about that. The core inference is just it has to get through the blood to get to these other organs in the first place. And if it's true that only what's getting into the blood is a tiny fraction of, at any point in time, of the normal aluminum level, this is not going to make a difference because you already have a normal aluminum level that's at least 100 times greater than what is getting into the blood at highest point.
Okay.
So to say it's going to be consequential would be absurd.
But we're also not testing amorphous aluminum hydroxysulfate phosphate. Correctly?
I knew that was going to come up. Thank you for mentioning that. I am so glad you mentioned that. I am so glad you mentioned that because that actually, the reason, and yep, so I anticipated this, and this was also mentioned in the anti-vax paper. So the issue with that is the amorphous. I get why they're trying to do it. They're going to they shoot themselves in their foot by mentioning that. They're trying to do that because they want to make the narrative that it's not eliminated rapidly. And they want to say, okay, yes, aluminum phosphate is aqueous. That's going to absorb quick and get removed, even though it was aluminum hydroxide that would get removed. But they have an explanation for that, the particulates in the injection. Fine, let's ignore that. They want to make the case that it's the elimination that's not going to be fast. Like, oh, you're saying it gets eliminated fast. It doesn't, because not necessarily, because it's a gel. It's an amorphous aluminum. So you would expect it to be absorbed slow. If anything, that would be even safer. If anything, you would actually have a safer profile because it would be absorbed from the granuloma into the bloodstream slower than definitely aluminum. And the only reason you saw a high initial absorption of aluminum hydroxide was because the needle ended up, the particulates, and this is even mentioned in the anti-vax paper, that the particulates of the needle ended up going in and out and may have nicked a blood vessel or a arterial. So this criticism, yeah, sure, it's a criticism, but again, it's a steel man. Looking at aluminum hydroxide or aluminum phosphate, if anything, would be a steel man in the unsafe direction. If we looked at the gel, it would absorb even slower. And I would make the same point even stronger.
Okay, I guess I'm at the point where I'm not convinced because okay. Do you agree that we have found aluminum in the autopsy brains of people with Alzheimer's, people with autism?
Okay, so I wish we could have gone through the data on aluminum in Alzheimer's. However, we did not post that to each other. If you want to have another discussion about that, we can, that's fine. I think it is a consequence of the disease. It's not a disease causer itself based on the normal levels of aluminum in the body. I think that's a result of the disease process, not a causal agent. But we can have a debate on that with data at a different time if you'd like.
Okay.
But that doesn't, again, notice that doesn't address the inference, though. This isn't addressing the inference. Even if aluminum was causative, even if it was causative to Alzheimer's, the problem is, again, that the area under the curve of aluminum that's getting to the blood from the injection is less than 1%. You would expect it to be, based on this data, less than 1% of the normal aluminum that's already there. You have to wonder how clinically significant that's even.
I'm losing you.
Oh, okay. What I said again, so can you hear me now?
Not too well. You keep cutting in and out.
Okay, hold on. Okay, so what I'm saying, let me know. I can try to change. Let's see here.
I can hear you perfectly fine. I think it's just.
I could join. I could try joining on my computer again. Let me know if that's okay.
Sorry, I didn't have my mute off. I can hear you perfectly fine. If there's any weirdness, it's not the stream's not hearing it. It's just going to be on Ben's end.
Okay, so it's on Ben then. All right. Look, Ben, all I'm saying is that the key point here is that if you're going to this problem, what you need to say is that based on this data, even if percent area under the curve difference is going to make it to the blood anyway, by this data, drawing the blood at multiple different times, showing the highest peak being less than 1% different. Even if you want to say that being less, you would have to say that this difference of less that wouldn't even be detected on a normal aluminum count, a normal aluminum level from this injection, is going to cause Alzheimer's or something. That is a huge claim. That is I don't know anyone who would stand by that claim. To the degree of a normal, it's within the range, well within the range of the normal aluminum level. It's like, here's the normal aluminum level. This is the little difference you're arguing about. Not even that. That's even an exaggeration. And I haven't seen any point to respond to that. What's the response to that?
So my response is essentially like, we have a serious accounting problem of where has the aluminum gone. And if it is still all, in fact, in a granuloma in the muscle, it should be very easy to get that out, measure it, and see.
It's actually not true. It's not easy. That's not easy to do. That's not necessarily easy to do because the granulomas could be very small. You see the granulomas on a microscope. You would have to section large sections of muscle just to find it. It's not the case that it would be necessarily easy to find. But anyway, the point that also doesn't address the point, Ben. The point is that regardless of the accounting, we know that at any point in time, the aluminum from the injection, at any point in time, none of the aluminum from the injection showed up into the blood at a level that is higher than 1% of the normal aluminum level. That subsumes all of these things because in order to get to any of these areas, whether it's excretion, whether it's the brain, whether it's the bones, it has to go through the blood anyway. Regardless of the accounting, that doesn't attack the inference.
Of course, the blood level is never going to get to a point where it's toxic, to like acutely toxic.
Not even that, Ben. Not even that. Not just that it's not getting to a toxic level. It's not getting to a level that's even 1% different than the normal level at any point in time. And by the way, the only reason it even got to a point of 0.8% was because of the nicking. They speculated, even the anti-vax papers suggested that they were nicking the arterioles. And after that, it was far lower at any point in time with the tracer. I mean, look, here's the thing. If you continuously monitor the blood, you wouldn't see a difference. You would see no difference. Unless you had some ridiculously special equipment that would detect like these insanely different differences. You would see no difference after an aluminum adjuvant vaccination. You just wouldn't, because its releases so slowly.
Yeah, but then it begs the question of like, how much needs to get to the brain for it to be toxic? How much needs to get to any other tissue for it to be toxic?
Sure, that's fine. But regardless, but the point is, if you're going to say that amount, if you're going to say that change in blood can end up getting to the brain and be toxic, that's true. That is true for normal aluminum levels. The point is that, look, it's a sea of aluminum. At any point in time in the blood, it's a sea of at least 100 aluminums for every one aluminum in there that's coming from a vaccine. And really, it's lower than that. Really, it's like, if that was only the initial point, after the injection point, which they speculate the nanoparticles got out, it's really probably like one aluminum, a vaccine aluminum that made it to the blood for every 200, 300, or 400 aluminums that are in the blood just naturally, just part of the normal aluminum level. That's the degree of how low you're talking about here. To suggest that if you suggest that that's what's going to cause a problem, you would have to suggest that literally just everyone's going to have a problem from the aluminum, because you're talking about such a small difference in aluminum that is so well within the range of the normal aluminum range. It's just absurd.
Yeah, but there's a lot of, this is where we get like super in the weeds about, I can't hear you, maybe you're still talking.
No, I'm not still talking, I'm good.
Okay, we're super in the weeds about what's going on mechanistically. essentially we can speculate, but we don't really know. Is that a fair point?
Well, I think we do. Well, I think we do really know, because I think we have the RCTs. But I'm saying even on the mechanistic data, I think we have enough of mechanistic data to suggest that it would be insane. It would be that for one aluminum for every 100 at max, one aluminum for every 100 making it into the blood at any given time, given what the normal blood range of aluminum is, would actually result in anything. And by the way, they did do autopsies on some organs. And again, the point could be made the same. They're looking at tracers. But again, if they included the normal aluminum, if they showed you what the normal aluminum was compared to that, it would be so obvious that there's no way this can make it. But again, I agree. We don't know based on the mechanisms. We put it to the test. On the mechanistic level, looks ridiculous. The RCTs, 1,000 and 500, nothing. You just have to keep raising the goalpost. You just have to keep shifting it around, raising it enough such that Gardasil isn't safe, vaccine's not safe. And that is the degree, look, that seems to be the degree of distancing from reality that we're seeing here. And I just want to hold you to consistency, because we can pick this up, and I'm going to show you the data for all these other medications. I can show you the data for all other medications for which you would say is not safe. I guess that's my closing statements. Yeah.
Yeah, it sounds like that's the one thing that we agree upon, that if you can show a similar rate of autoimmunity for other drugs, then that's where I guess we disagree.
Based on the criteria of how that autoimmunity was counted. Again, it's not going to be hard, because based on how this was tallied, you're going to find it. So we can schedule up a follow-up debate, and I can get you biting the bullet on just a whole bunch of litany of other commonly accepted to be safe medications, and you're just going to come out saying, okay, well, everything we can't say is safe. You can have that, but I just want you to just examine, honestly, just examine your internal epistemic standard of just, how did I get here? Position where I'm going to end up biting. So, yeah, I mean, you're either going to be your position for anti-vaxxers, you're either going to be yourself.
Avi, did you change your Wi-Fi
you're going to be
or
in an absurd
something?
position.
Your sound.
You started
Avi,
off
did
in
you
logically.
change your Wi-Fi or something? Your sound is just cutting out like crazy.
I did not. Oh, sorry, it just came on. Sorry.
Oh, okay. There you go.
Sorry, can you hear me now? Hello? Hello?
Totally lost.
Can you hear me now?
Yep, loud and clear. Sorry, I just have to double unmute myself to get to voice. Yeah, you're good.
Okay.
Yeah. So my position is for anti-vaxxers is that you're either in one of two positions. Yeah, go ahead.
It sounds like we're ending it where you have to show me that 2.3% coincidence with administration of other drugs is kind of par for the course with autoimmunity. Is that right?
Based on how that is tallied, yes. Based on how that was counted, I agree. Yeah. If you count it the same way, if you take all of those conditions that are listed and you summate them, you will see 2.3%. It is not going to be hard to see 2.3% for those conditions. Yeah.
I think it's so you show that basically because in the general population the incidence of these is higher. Not because the medications cause them, because when you tally it like that, just these are very common things. Like arthralgia is just very common. You're going to see at least 1.1 in one group and 1% in one group came from just arthralgia, arthritis, and arthropathy alone. You see these like writer's arthritis, these are arthralgias in general, arthritis in general, arthropathies. These are just common things. You're already going to cut that down to 1.3%. Other things are also common. Psoriasis, so common. Pigmentation disorders, fairly common. And then when you just summate so many other things up. It's not an apples to apples comparison to just look at the rates in the general population. We're looking at coincidence with administration of a drug.
Yes, no incidents. Yeah, no, but that is what it is. AEs are just an incidence of something that happened or a new medical condition that developed after administration of a drug. Sure. The reason you're going to find it is because that incidence itself per year is really what you would expect anyway. So you're going to find this with just about any medication.
Okay. I mean, I think, we could say this, but like, I just, when it's coincidental with the drug, I don't think you're going to find it for other medications. I mean, I hope the data's out there and we can look at it.
And if I showed it to you, then what? What happens? What if I showed it to you, and it turns out that it is true that you see these things with if you tally it accordingly, you do see 2.3% or higher. What then?
Yeah, I would be concerned.
You would be concerned about what?
For the drugs that showed that this 2.3%.
Let's say it's every drug. Let's say it's every drug, or just about every drug. And what I'm getting at here, Ben, is there anything here that would get you to say, hey, wait a minute, instead of saying, well, I'm concerned about the drug, is there anything that would get you to say, hey, I'm concerned about my epistemic standard? Maybe it's just my standard that's a little crazy here. Maybe I should reevaluate and say,
for every drug to show the similar profile, then yes, I would concede my argument.
Okay. I don't know if I'll be able to do it for every drug, but I think in the vast majority of cases, again, like the reason you can do this, you should be able to do this because this is well within, if not lower than the prevalence of the yearly incidence. Sorry, the yearly incidence within this age group anyway. So, yeah, I mean, based on how it's tallied, it'll be able to be shown. And so I'm glad that, okay, if you're shown this, just to be clear, drug after drug, you end up finding this. You would have to reevaluate your epistemic standard.
Yes.
Okay, cool. All right, I think we could end it there. We can do you want to do closing statements?
Sure. First off, thank you for hanging with me for 4 hours and keeping it respectful. I really appreciate that. I guess, I don't have much to say in terms of a closing statement. Just that this 2.3% is alarming to me. I think that there are ways that you can, essentially look at the data a certain perspective that would show you something that it's not an apples to apples comparison. So I await to what you have to say. And I know that the incidence of autoimmune disease is something like, depending on whom you ask, 1 in 7, 1 in 14. But when we're looking at it in terms of age, people aren't born with these autoimmune diseases. It happens later in life. Mine didn't happen until I was age 27, 28. Or as far as symptoms. I think that that data will vindicate my viewpoint, but it's up to see. And I guess originally my whole assertion all along was that you don't really have the data to know whether it's safe or not, and people should make their own opinions. So I hope that if you stuck around for the whole thing, you learned something, and you had more data than maybe you had previously. So okay.
Well, before I give I hate to respond to closing statements. I don't like doing it, but just the one thing I did notice, Ben, is that on your Instagram you said something different than that. You said that you had unequivocal proof or something to that degree. That
okay, fair enough. This is unequivocal proof. The package insert, but maybe I'm wrong. Okay, so
just to be clear, you don't think your statement that this is unequivocal proof that these vaccines cause in autoimmune conditions, you reject that?
I would have to say that I have unequivocal proof that they make me suspect that they do.
Okay, that's a different statement. Okay, so you don't. It's not the case that you have unequivocal proof that they cause autoimmune conditions. Okay, so I'm glad you rejected what you said previously. Okay, cool. In terms of my closing statement, look, there's an evidence hierarchy. Mechanistic speculation is on the bottom. randomized control trials are on the top. Meta-analyses of randomized control trial, I would put even, if done all things being considered equal, I would put them on top of that. In this case, we have at least a sample size of 1,500 in a randomized control trial, 2-to-1. We have clear data. In that data, it was a complete wash between Gardasil and the placebo. We saw nothing. The SAEs that we did see, it was clear in the majority of those cases that they were not even from the vaccine. SAE rate of 0.16 is all you could really say. As far as safety goes, compared to other safety data, based on even looking at how the autoimmune conditions are tallied, within that age group itself, you wouldn't expect to see any difference. And as far as drugs go, based on this data, it is as safe virtually as safe as it gets. And I will say, from my view, from my standard, unequivocally, to whatever degree, if you want to say that Gardasil, you can't say it's safe, you will be either in a logically contradictory position, or you will be forced to bite the bullet on virtually every medication being not safe by the same standard. All right, and then with that, thanks for watching, thanks for joining, and I'm more than happy if there's any, we can pick up the conversation of the other data at a later time. I'm more than happy to accept challenges from any other anti-vaxxer. I know some anti-vaxxers are offering money. Happy to take that as well. So I know there's a lack of physicians that are willing to step up to the plate and debate anti-vaxxers. I'm not one of them. I'm happy to debate them and happy to provide the information to the public. And yeah, thanks for watching, guys. And sorry for the technical difficulties. I do have some better equipment coming in the future. So.
All right. Good night. Thank you.